patients. We have chapter groups dedicated to rare forms of myeloma or related diseases like amyloidosis, non-secretory myeloma, and plasma cell leukemia. We also have chapters for African Americans and patients that speak Spanish. Forming these groups creates a powerful bond, a larger network, and incredible research opportunities as we join together virtually and occasionally in person once a month. It's a privilege to get to know so many myeloma patients, caregivers, physicians, and supporters intimately and support them in their journey. I absolutely love my job.
HealthTree Myeloma Roundtables are live patient education events featuring top myeloma experts. They're designed to provide in-depth and accessible overviews of the landscape of particular myeloma issues for novice and veteran patients. Myeloma Roundtables teach about particular topics in myeloma, not just basics. They help put things into context for patients to learn and understand more about their disease and how to best live with it until cures are achieved. Whether you're newly diagnosed or relapse refractory, want to learn about myeloma testing, genetics, side effects, or the latest research in cutting-edge therapies, Myeloma Roundtables are for you. Each has moderated discussions, and the best thing I think about our Myeloma Roundtables is that they always have extensive time set aside for the audience to ask questions of the full faculty. This allows for interaction among the speakers and often provides insights that can't be found in papers and studies. Our goal is to help you ask the best questions so that you can have more productive visits with your healthcare team. We host 6 roundtables each year throughout the nation, and we live stream, record, and post each event on our website. We call them roundtables because we believe everyone touched by myeloma, patients, caregivers, family members, researchers, physicians, nurses, and anyone else who comes into contact with myeloma patients deserves a front seat at the table when it comes to the best diagnosis, the best treatment, and a realistic path to a cure.
Whether you are a caregiver or a myeloma patient, the HealthTree Myeloma Coach program is designed to help you find one-on-one support to fit your needs. You might be looking for emotional support, more information about myeloma, or just someone to talk to. No matter what you're looking for, coaches are trained volunteers and offer their personal experiences and resources to help. You can find a coach in your location or with specific areas of experience that would be helpful to you. You can connect in a way that works best for both of you, by phone, email, video chat, or in person. You might even want to become a coach yourself to help someone else on their journey. Orientation and training are provided to the coaches using our extensive HealthTree University curriculum, so answers to common questions are never far away. It's the personal experience that sets HealthTree Myeloma coaches apart. Having myeloma can feel so lonely and isolating. When my husband was diagnosed with myeloma over 7 years ago, I wanted to talk to someone who was close to my age, balancing children at home, working, and caregiving, to learn how they did it and what had helped them. I wanted to talk to someone who could personally relate to what I was experiencing. The HealthTree Myeloma Coach program can provide these types of connections. Knowing there are others who can relate can make all of the difference. I love leading this program because it truly helps us meet people where they are, provide personalized resources and support, and shows patients they can live a happy and full life, even with a myeloma diagnosis.
Educated and empowered patients tend to have better outcomes. HealthTree University for Multiple Myeloma is designed as an online myeloma curriculum to meet all myeloma educational needs. Whether you are a newly diagnosed myeloma patient or a myeloma patient expert, HealthTree University includes dozens of online courses with hundreds of lessons. Each course consists of multiple video lessons focused around a single topic. These lessons are taught by the myeloma experts who are world-renowned and treat hundreds of myeloma patients each year. They are the leading researchers in the field, with their findings being published in well-respected medical journals. The lessons also include video animations and graphics to help make difficult concepts easier to understand. Quizzes follow each lesson to evaluate the understanding of the topic. HealthTree University is continuously updated as the myeloma landscape changes. We are constantly interviewing myeloma experts at our roundtable meetings and key myeloma academic meetings to keep the curriculum up to date. When I was diagnosed with multiple myeloma, I believed in Doctor Knows Best, and I blindly followed my doctor's orders. As I've become more experienced, and as a former teacher, I realized the importance of arming patients with the information in HealthTree University. I want you to make informed decisions and become your own best advocate.
HealthTree's Black Myeloma Health Program is aimed at increasing awareness, education, outreach, and advocacy in our Black communities. I understand the need personally as a myeloma caregiver to my husband. We offer help and support for those who are on this myeloma journey, a blood cancer that African Americans are 2 to 3 times more likely to develop. We also have a higher incidence of the precursor conditions MGUS and smoldering myeloma. Our programs aim to promote awareness, education, outreach, and advocacy to this patient group. We are doing this by creating a blackmyelomahealth.org website, teaching the Black community about the signs and symptoms of myeloma, taking our programs and events into the Black communities where health and wellness education is perhaps not free-flowing, partnering with Black organizations to perform greater community outreach and awareness, growing our African American Facebook group and HealthTree community chapter, and creating education in HealthTree University for the Black community. Taken together, we strengthen our African American communities and we effectuate positive change.
The HealthTree podcast for multiple myeloma was our very first program that connects patients with myeloma researchers. In the podcast, I interview top myeloma researchers about key myeloma topics and about clinical trials. Finding and joining clinical trials is not an easy task for patients. We need to understand what trials are open, who can join, and which patients are the best fit. So to date, I've interviewed over 150 myeloma experts to connect you with their open clinical trials. The program's been viewed or listened to by over 2 million readers and listeners and continues to be one of our most popular programs.
Exercise has many benefits for patients, including reduction of fatigue, decreased fall risk, improved mood, and overall better quality of life. One of the most important benefits for patients is that being fit can maximize the number of treatment options available to you. Our HealthTree Moves program was created to build community and increase movement. Throughout the year, we have a range of fitness challenges in our mobile HealthTree Moves app that is designed for all fitness levels with the goal to stay accountable and continue or increase activity. We have found through these challenges that participants are motivated to hit the goals they set for themselves because they have a group of others to do it with. We are also expanding our HealthTree Moves program with a growing 5K and 1 mile series, so watch in your area for an upcoming event. Our goal is to bring awareness, build community, and get moving together.
HealthTree Cure Hub is a patient tool that helps you navigate your myeloma and at the same time accelerate a myeloma cure. We built HealthTree Cure Hub to provide you with easy-to-use, everyday tools that will help you make better treatment decisions in partnership with your healthcare team. In Cure Hub, you can track all your myeloma labs in a single place so you don't have to create or maintain a separate spreadsheet. Cure Hub can help you look ahead and find a list of treatment options personalized just for you that you can consider based on your prior therapies, genetics, and other factors. HealthTree Cure Hub can also search through hundreds of open myeloma clinical trials and narrow the list down to only those you are eligible to join. You can find and connect with patients that have similar experiences or genetics using our Twin Machine technology. You can also use our side effect feature, where you can see hundreds of side effects and thousands of crowdsourced solutions provided by patients. Learn from your peers to see what worked and what didn't to manage the most common myeloma side effects. I encourage you to sign up for HealthTree Cure Hub and investigate all the tools provided to help you make the best decisions possible.
More than 11,000 patients have joined HealthTree Cure Hub to contribute to research aimed at accelerating the cure for multiple myeloma. Because so many patients like you have contributed your myeloma story, we can now use this real-world data to reach faster conclusions and answer key questions such as, what are the best induction therapies? How are patients making treatment decisions at relapse? What are the best treatments for specific genetic subtypes? What are the outcomes of novel treatments like CAR T therapy?
Patients have pressing health questions. Therefore, we need a practical way to help inform these key treatment decisions today.
Our HealthTree for multiple myeloma research program partners with investigators to answer critical medical questions.
We facilitate the ability to run queries on anonymized data and enable investigators to ask you questions.
We would like to thank you for contributing and responding to these surveys and participating in the research.
Because when you do, you improve clinical care in the immediate future.
If you haven't already, please join us in HealthTree Cure Hub to contribute to answering the critical questions myeloma patients and researchers are asking today, and to help us drive faster to a myeloma cure.
As we identified the tools you needed to successfully navigate your myeloma, we realized that third-party software was not going to be ideal. It was too disjointed and it didn't work the way you wanted it to. We have now built over 10 integrated software tools to support myeloma patients and caregivers. Nothing like it exists for any other disease, anywhere. To make it easier for you, each tool is part of a software suite where you can create one account using single sign-on. We use the latest technology and security methodologies to ensure your information is safe. Our development team of over 12 members ensures that we will continue to support you as we add new products and services that we roll into the HealthTree platform. We listen and respond to your input, so please feel free to share your thoughts so we can make the platform even better over time.
HealthTree for Multiple Myeloma has a diverse team of patient navigators who are always happy to help by phone or online chat. For newly diagnosed patients, knowing where to start can feel overwhelming. Our team can help you understand your disease, your type of myeloma, and suggest questions you may want to ask to your care team. We can help connect with a myeloma expert and explain HealthTree resources that can help you navigate your individual situation. For relapsed and more experienced patients, we can guide you through resources you may have not known existed. For example, understanding and keeping track of your myeloma is an important way to better advocate for your own care. Our team provides support for HealthTree Cure Hub, a tool that helps you navigate your myeloma while also helping move myeloma research forward. Our medical experts can help you gather all of your medical information into a single place, even for multiple facilities. This helps you keep an eye on your myeloma markers and track your key labs so you can more easily watch for relapse and ask better questions in the clinic about your next potential therapy. Our team includes African American patient navigators and Spanish-speaking patient navigators to provide more personalized support for those who may need it. Let us help you prepare for upcoming clinic appointments or better understand your disease by calling 800-709-1113. As always, we're happy to help.
For more information about our programs, visit healthtree.org. Welcome to today's HealthTree Roundtable for Multiple Myeloma. I'd like to introduce Jenny Ahlstrom, founder and CEO of the HealthTree Foundation, and a myeloma patient herself. Here's Jenny.
Ready? I'd like to welcome you today to our third roundtable in Buffalo, and we also have an online audience for both of the sessions today. Because of the pandemic and everything, we do have a few restrictions. As you can see, they ask when we're in patient areas to keep the masks on the campus, in the buildings in the campus, I should say. You saw where breakfast was, and that's where lunch will be as well, and we'll take a break there. To start out our day, I'd like to introduce our founder and are you chairman or president? I don't even know. I've never thought of that. CEO. Well, she's our founder, and we wouldn't exist without her. Let's put it that way. With that, I'd like to introduce Jenny Ahlstrom.
Well, hi everyone, and thank you for coming. I'm not sure if I know how to advance the slides. Oh, yeah, I do. Okay, thank you so much for coming. I'm so impressed. We love Jens and are so proud of him and what he's built in this program. And we're so grateful for the others like Dr. Maury Gertz. You're going to love his presentation, and Irvi Shaw and others who are speaking today. So you're in for a fun day. If you retain even a small percentage of what you learned today, it will be a successful day. And so just be patient. If you're in the newly diagnosed category, it's a lot of information to pull in all at once. But first, I would just like to thank our sponsors who have tables outside. So feel free to stop by their tables and chat with them as you have lunch and want to mingle and learn more about what they have to offer. But we'd like to thank Takeda Oncology, Bristol Myers Squibb, Janssen Oncology, Sanofi, GSK, Amgen Oncology, Karyopharm Genentech, and Legend Bio for supporting this roundtable program. We're really grateful. I just wanted to give you a little bit of an overview if you're not familiar with some of our programs. We've shifted, you might know us as the Myeloma Crowd. We're shifting our name over to HealthTree. And part of that is because when we came out with this HealthTree tool that I'll talk about, we started confusing everyone, like, what's Myeloma Crowd, what's HealthTree? And we kind of needed to make a decision or commitment one way or another. And people seem to like the HealthTree name better. So we're calling it HealthTree for multiple myeloma. And here are some of this is our vision, our mission, and our strategy. And I'll just kind of go over a little bit of our programs. So in summary, this is what we do two things primarily. We support community through a news site, a community site, a podcast, these roundtable meetings, a specialist directory. We have coaches that will talk about a fitness app and a university. We also have, and then we do another thing which is we help accelerate research. My myeloma journey didn't really start with my own diagnosis. I was diagnosed in 2010, but 6 years prior to that, my brother-in-law had developed AML. We had this, it was a crazy time. He was quite ill and he lasted a year. AML is a very serious cancer and very acute condition. And through that experience, we learned we had this hypothesis: what if patients would share their information together? Could we as patients advance research faster? And the answer to that is yes. And now we're doing it in myeloma. So we're super excited. Some of you may have come to our workshop in 2018 when we did that and learned more a little bit about that. So we have a portal called HealthTree Cure Hub. It is a patient data portal where we can share our myeloma stories with one another. And we have 10,500 patients who have joined that portal so far. And we can help accelerate research. We're doing that, and I'll talk a little bit about that. But we're doing research with actually most of the speakers today on that. And that will include a clinical trial finder as well. This is our team. So we have grown. It's not just me anymore. In 2013, it was just me. And then we added on the roundtables and a coach program. And now we have an extensive team. That includes patient navigators who are available by phone all the time for you, and those who will validate kind of patient profiles. These are our advisors, and we're grateful to them for helping us. And then this is the news site. So if you go to healthtree.org/myeloma, you'll see kind of a whole platform of tools that we have. It's not Myeloma Crowd anymore. I should have changed that. And last year we had over 850,000 unique visitors to the website. So if you're looking to stay up to date on myeloma news in a friendly sort of way, this is a good way to do it. We make sure all our articles are understandable for the lay patient, because not all of us have these, extensive degrees that we can understand the scientific research papers. We now have a video journaling option. So if you're looking to record your myeloma story, you can do that by video, audio, or in text. It's a really nice way to remember, like, how was I diagnosed and how did I tell my family, and some of those personal things. You can keep that private, or you can share certain posts with your family on social media or by a link or by email. So that is now live. That went live last month. We have something called HealthTree Community for multiple myeloma, where we have chapters and chapter events. And those are online webinars. Last year, we hosted 136 webinars. This year, we might almost double that. And we're targeted to have about 113,000 patients watch or listen to those webinars. So you can join a chapter. There's like an immunotherapy chapter, or there's a relapse refractory chapter, or there's a newly diagnosed chapter, there's a fitness chapter, there's a nutrition chapter where we've posted a lot of Irvi's webinars. So it's that's our community program. And we're starting also, we have now a Northeast chapter, so you can join that chapter if you'd like. And yeah, I think Beth is part of that chapter. This was my first program, which was podcasts with experts. I've now done 156 interviews with investigators like Dr. Gertz and Dr. Hillengass. And it's a 1-hour podcast. And it's a wonderful way to listen in about a clinical trial or a topic in myeloma. And we try to help. This came out of me wanting to join a clinical trial. And I called 8 facilities. I got a call back from 2. I started reading on clinicaltrials.gov. It was sort of a nightmare. And I thought, no wonder patients don't join clinical trials. It's so hard to understand, like what's it all about anyway? So that's what that podcast is aiming to do. We do these live meetings. You're at one. We have a coach program. We have over 200 coaches, and coaches can help, especially for newly diagnosed patients. They have topics that they can coach on, like if you're taking a certain drug or whatnot, or you're going to go through stem cell transplant, or if you need financial help or any of that. Beth, where are you? She might be outside, but Beth is a coach in that program, and it's wonderful. So we have a coach summit every year, and we've coached over 1,500 patients. So you get to choose your own coach. You can choose a coach for, a month or 6 months or longer, and lots of wonderful friendships have developed through this coaching program. And we have something called HealthTree University, and that's one of our most popular programs. And at meetings like this, we'll pull the doctors aside and say, hey, could you answer a few basic questions or complex questions? So HealthTree University now has over 41 classes and over 600 video lessons, and they're organized by topic. So when I was diagnosed, I went to a meeting and picked up a little, and went to something else and picked up a little, and read online and picked up a little. It really took me like 6-8 years. Some of us do not have the luxury of that kind of time frame. So we've had newly diagnosed patients say, oh, I binge watched my way through HealthTree University. So I would check that out. Anybody can watch it. You don't have to be a patient. You can be a family member, friend, caregiver, whatever. And then this is the HealthTree Cure Hub portal that I talked about. We did a little workshop yesterday. In 2018, we kind of did a 50-city tour. I think we slept in our beds all of 5 days. With my family, my husband and 4 of my kids came with us, and we just listened. So this is a way that we can help accelerate research, but it also has benefits back to you. You can see treatment options personally relevant to you based on where you're at. You can find clinical trials. You can match with a twin. You can see side effect solutions that other patients have used. You can participate in surveys and studies. We'll be doing a survey on amyloidosis with Dr. Gertz because these patients get a late diagnosis, and we want to understand how are they actually getting diagnosed. So we're doing that with him. And with Dr. Hillengass, we've supported some of his fitness studies, and we'll continue to work with him on that. Dr. Shaw, we're working with her on her nutrivention studies, and we will actually be facilitating one of her studies to see if progression from smoldering myeloma to active myeloma can be slowed down based on diet. So it's those types of things that we can do. Nathan will be talking a little bit later on that, but just so you're aware, you can have a profile and then help us answer questions. We're able to, and Nathan will talk about it a little bit more. So we have a clinical trial finder that is now live. on our site, and we will get it a little more personalized, but now we can do advanced filtering. So you could find all the clinical trials. Like I want to find a phase 2 CAR T trial in my area and get on a waiting list or something like that, or a bispecific antibody trial or whatnot based on your search criteria. We have a fitness focus because it's really important for myeloma patients to stay as healthy as possible. We have a HealthTree phone app. So if you want to join that app, it's on Android and iPhones, and you can just download it. It's called HealthTree Moves. You can join fitness challenges that we do, like for the month of September and March. We do a 30-day fitness challenge, and you can just go walking or swimming or whatever you want to do, track your minutes. And we're building out a 5K series, and we just did a Black Myeloma Health virtual 5K. We now have a program specifically for African Americans and a program for Spanish speakers. And so we were working a lot on these disparities kinds of things. And that's really what I wanted to share. So thank you so much for your time.
I'm glad, I know Jenny's feeling under the weather, but I'm really glad she was able to share that with you. Our next speaker is, I'll be honest, is my favorite speaker. He's spoken at more of our events. I guess that's the proof of the pudding of over the years of this program than any of them. And I think he's arguably the most engaging person. People always ask me, who would you go see if you were diagnosed with myeloma? Because I've been working, for a number of years, and I was telling, well, I would go see 3 people, and I won't tell you who the second and third are, but I would go see Maury first, Dr. Gertz first, because he could explain to me what's going on better than anybody, and then I can start making decisions after he let me know what I had to deal with, and that's why I like to have him come to these events. So with that, I'd like to introduce Dr. Maury Gertz from the Mayo Clinic.
Oh, that's good. So, the history of chemotherapy we need a mic for the online audience. I have to hold this? Yeah.
We also have one for you.
Fine. This compound, ClCCSCCCl. Got it. So this was developed about 1910, this sulfur-containing compound. And when it was first used, it was used on the battlefield in World War I. And when it explodes and you're exposed to it, it causes amazing severe irritation of the respiratory tract, the nose, the throat. The irritation is so profound that it causes battlefield asphyxia, and the recruits die on the battlefield. Now, this bischloroethyl sulfoamine is technically difficult, but when it explodes on the battlefield, it's bright yellow. It's the same color as the top of this sheet here. So they referred to it because it's just easy to identify as mustard gas. And so mustard gas was used to asphyxiate, against the Geneva Convention, troops on the battlefield. But not everybody exposed to this died of it. And what happened is that there were some military recruits who would survive the initial damage to their lungs for 2 weeks. And then at the end of 2 weeks, they would die of infection, bleeding, and although it was very limited, you could see that these patients had very profound anemia. Military recruits who die on the battlefield, you don't need permission for postmortem examination. So they routinely underwent postmortem examinations, and it was found that their bone marrows were completely empty, and their lymph tissue, the lymph nodes in their body, in the neck, chest, abdomen, were gone. They were just completely depleted from the exposure to the mustard gas. And it was thought that must have therapeutic value. And of course, this is 1916 now. Bone marrow technique hadn't been developed until the mid-40s. And so they thought there must be some value. And so the Department of War, which is what it was called in those days, before the Department of Defense, hired pharmacologists and hematologists. There weren't really hematologists, there was people interested in the blood, to try and make something out of it. And you couldn't handle mustard gas. It was impossible to handle a gaseous element. But what they did is they changed the sulfur to a nitrogen. And then they referred to it as nitrogen mustard. And that chemical, it turns out, nitrogen mustard, is capable, if you take DNA, which is that double helix, and unwind it and flatten it, it looks like a ladder. And the nitrogen mustard actually would hook up the uprights of the ladder. C, C, and C, C. So that it couldn't unzip. And if you can't unzip DNA, a cell can't divide. And so it would poison cells trying to divide. And so they said, okay, we've got something here that we can use to kill cancer cells. Well, they had the whole thing developed about 1943 and wanted to publish their results about the first anti-cancer drug in 1943. But the Department of Defense labeled it as top secret and blocked publication of the article. So it didn't come out until September 1946 in the Journal of the American Medical Association. And there are pictures there of people with very, very large lymph glands 10 days later, all gone. So they published this use of this nitrogen mustard compound for the medical professionals in the audience. I don't know if Jens is here, but the two pharmacologists who developed it were Goodman and Gilman. The two hematologists who developed it were Damashek, who was the first editor of the journal Blood, and Max Wintrobe, who founded the center in Salt Lake City. His picture is going to be in there somewhere when you go in. But nitrogen mustard was difficult. Just like the mustard gas could cause respiratory burns, if you injected this intravenously and it leaked out of the vein, it would burn a hole in your hand. It was extremely caustic and difficult to use. So they wanted to try and find something that was a little bit easier to use. So I'm going to draw the mustard here. Boom, boom, boom. And what they did is they wanted to make it easier. So they wanted to attach it to something that would get into cancer cells quickly. And so they wanted to attach it to an amino acid. Amino acid's the building block of protein. So amino acid, let's see, that amino acid that I'm drawing here is the amino acid glycine. This is the amino acid alanine. And then they attached it to a chemical group called phenyl. And that was phenylalanine. And when they attached it like this, that became L-phenylalanine mustard. Oh, that's not good. As long as I don't fall on it. So L-phenylalanine mustard, or LPAM, was introduced about 1960. And what they did, they renamed it generically as follows: MELPHAN, or melphalan. So that's been around 60 years. And melphalan is the drug that everybody gets in the context of a stem cell transplant, is melphalan at a very high dose. And that really is basically a derivative of mustard gas. It's not too far off and poisons the DNA. So that was the very, very first available drug for the treatment of multiple myeloma. It was actually produced in Russia, and it was introduced at 2 sites in the United States, or in North America. Dr. Daniel Bergsagel introduced it at the Princess Margaret Hospital, and it was introduced at the MD Anderson Hospital, those 2 by pioneers in the field. So that's 1. Drug number 2, it's the mid-50s, it's Germany, a lot of pharmaceutical companies, and they're trying to deal with the terrible problem that pregnant women get with nausea and vomiting. The technical term is hyperemesis gravidarum. And so they tried to produce medicines that would be both sedating and anti-nausea. And they did develop a compound, a very simple compound called thalidomide. So thalidomide came out in 1955, and they tested it in mice and in rats and found not to be toxic. But mice and rats don't have the same protein content as humans do. Now, thalidomide never got approved in the United States, never, because it caused peripheral neuropathy, and the FDA said, you can't sell this drug in the United States. It's too dangerous because of the peripheral neuropathy. But it was sold widely in Canada, Europe, Australia. And thalidomide caused terrible birth defects. And so it was basically banned worldwide and became available only available for the treatment of a skin condition that's associated with leprosy. So it wasn't a big seller. But in 1999, scientists looked at medicines that could stop tumor growth by blocking the growth of blood vessels that feed the cancer. And they found that thalidomide had this property of being able to stop blood vessel growth that feeds malignancies. And so the wife of a cardiologist knew this, and she brought this information to Dr. Barthel Barlaghi at the University of Arkansas Medical Center, 1998. And he said he'd try it. And in 1999, at the American Society of Hematology, he presented his results. I'd say there were 4,000 people in the audience. And when he showed the results of giving thalidomide to patients with multiple myeloma, you could have heard a pin drop, because it was really the first new medication for the treatment of multiple myeloma in 35 years. Thalidomide isn't used much in the United States, but its very, very simple derivatives, lenalidomide, also known as Revlimid, and its derivative, pomalidomide, also known as Pomalyst, are widely used as oral drugs for the treatment of myeloma. So that's 99. So we go to 2001 now. 2001, there's some poor MD, PhD at the University of South Carolina who's looking at drugs that might take care of cancer cells. And so he was looking at drugs that would inhibit what's called the proteasome. So the proteasome in a cell is basically the sink garbage disposal. And when proteins have to be recycled in the cell, they head to the garbage disposal and they're taken away. And so he tested this compound. Yeah, this would be about 2003 now, in a whole host of cancers. Breast, no activity. Colon, no activity. Lung, no activity. Pancreas, no activity. Treated 3 patients with multiple myeloma, and the responses were dramatic and deep and very, very impressive and very sudden. So the drug contains boron in it, the chemical element boron. The problem with boron is it damages nerves. It can damage nerves in your feet, and that's a problem with it. But That was part of the activity. And it was a inhibitor of the proteasome. And so they ended up calling it bortezomib, sorry. And that trade name for that's Velcade. The person who developed it is Robert Orlowski. And Dr. Orlowski is on the board here of HealthTree. He's now running the myeloma show at MD Anderson Cancer Center now. Bortezomib was number 3. So that's the third class, alkylating agents. Then secondly, were the immunomodulatory drugs with the thalidomide and Revlimid, lenalidomide, and then bortezomib or Velcade. Finally, I just want to talk briefly about scientifically targeted therapies, because those 3 were basically found by accident. All cells in the body have proteins on them, unique to those cells. Paul Ehrlich called those proteins antigens, about 1890. And antigens are familiar to you in a way because you know there are unique antigens on your red blood cells. Karl Landsteiner in 1902 described the unique antigens on red blood cells. The first one he found, he called A. The second one he found, he called B. If you had both the antigens, he called it AB. And if you didn't have those antigens, he called it O. But he didn't refer to it as red blood cell antigens, he called them blood types. But basically, that's the principle, unique proteins that identify cells. Well, there are unique proteins that identify myeloma cells. There are 2 I want to talk about. One of them, unique, is BCMA. BCMA stands for B cell maturation antigen, not important, is a protein found on myeloma cells. And what's nice about it is it's found only on multiple myeloma cells and no other cells. And so what scientists began to do, this would be just 7 years ago, is they took white blood cells, your white blood cells, killer white blood cells, as a matter of fact, the white blood cells that kill viruses and kill fungus that are really key to your immune system. And these killer white blood cells, oh, that's bad what I just did, are called T cells. The T cells refers to embryologically where they come from. And what they did is they wanted to activate these T cells so they would seek out and kill myeloma cells. So what they did is they engineered these T cells. And what they did is they put scaffolding on the T cells like this that was hooked in to the cell membrane with a recognition site here for the BCMA antigen. And so once it's engineered, your white cells, they can actually find and attack any cell that has BCMA, but the only cells that have BCMA are myeloma cells, and they'll kill these cells. Now, the engineered white cell, once you put that scaffolding on it to recognize myeloma cells, it's not a natural cell anymore. It's been constructed, and something that has pieces of nature in it, but isn't really natural, is referred to as a chimera. Goes back to the Iliad. Homer described it. There was an animal that had a lion's body and an eagle's head and a snake's tail. So they created this chimera, and this recognized the BCMA antigen on the myeloma cell surface. So it was an antigen receptor T cell, or CAR T. And so CAR T therapy was specifically designed and engineered to directly attack multiple myeloma cells. We have 2 CAR T products approved in the United States after failing 4 treatments, but there are a lot of research CAR T trials. I mean, a lot, including a trial now called Cartitude, where patients actually start treatment for their multiple myeloma, and instead of having a stem cell transplant, they actually are randomized, a coin toss. Half get a transplant, half get CAR T therapy. The goal to determine, will CAR T replace the use of stem cell transplant? The second iteration of this is once they developed this scaffolding that was hooked in to the killer T cell, your white blood cell, and then would recognize the myeloma cell, would say, well, why do we have to engineer the white cell? Why don't we just produce a molecule that on one end recognizes the killer T cell, and on the other end recognizes the myeloma cell. So it will attach at both ends, but it doesn't have to be engineered into the white cell. And this is what's referred to as a bispecific 1-2 antibody that at one end recognizes the cell that kills and number 2 recognizes the cancer cell, brings them together. And so bispecific antibodies. I'm going to finish. BCMA is not the only myeloma specific cell. There's another antigen on the surface of myeloma cells called CD38. CD38, again, is a protein that's found only on myeloma cells. And so they specifically, this would be Janssen actually, specifically engineered an antibody to recognize CD38 on the myeloma cell surface and kill it. And when it was first introduced, maybe experimental trials 2014, 2015 in heavily pretreated myeloma patients, it was found to be highly active in killing the myeloma cell because the antibody recognized the antigen, the protein unique to myeloma cells called CD38. And they called this monoclonal antibody daratumumab. You know it as Darzalex, as the 5th category of chemotherapy. So 5 categories: alkylating agents, mustard gas, immunomodulatory drugs, the ones that cause birth defects, but in humans, not in mice or rats, proteasome inhibitors, the proteins that break the garbage disposal, so the garbage collects in the sink and kills the cell, CAR T, the engineered white cells to recognize specific BCMA proteins on the surface, and 5 monoclonal antibodies against CD38, of which daratumumab is one. There's a second one, isatuximab. I don't know what the trade name is for isatuximab. I can't remember. Sarclisa. Thank you. And so those are basically the 5 drugs. So I just wanted to kick things off by giving a little bit of an overview of the development of the chemotherapy drugs for multiple myeloma. And Greg, you can take it away from there.
So we're going to do a little, we need to get the screen down and we're going to move this. And we'll pay you a little extra, Dr. Gertz, for that. One of the things that you should know, Dr. Gertz mentioned at the beginning of his talk, he mentioned a Dr. Damashek, who was one of the founding fathers of hematology in the United States. Dr. Damashek's one of Dr. Damashek's star pupils, and for our purposes, the most important star pupil, was Dr. Robert Kyle, who still works at the Mayo Clinic and now in his 90s. And he basically invented everything we talk about, and he defined everything we talk about in this. And so Dr. Kyle was a direct protege of Dr. Damashek. Dr. Gertz is a direct protege of Dr. Kyle. So you are kind of like being at a physics lecture, you know, listening to the third generation of Albert Einstein. And you can understand why I like to have him here. He puts everything into context for you in a way that's really understandable. You'll hear a lot of these concepts again as the day progresses, but we wanted to give you a quick background on that. If there were a Mount Rushmore of myeloma experts, certainly Bob Kyle is inarguably on it, and Bart Barlegi is inarguably on it. And there would be a debate about, you know, who's the first and second tier for the next group. One of the names that would definitely be in the conversation is Dr. Philip McCarthy from right here in Roswell Park. As you know, stem cell transplant is a major issue in myeloma. It continues to be. And there is probably no better expert in the world to talk to you about the context of stem cell transplant and how it fits into your treatment. So with that, I'll introduce Dr. Phil McCarthy.
I am not Dr. Philip McCarthy, which you can hear on both on my accent as well as on my looks. I have to, sorry. Yeah, I have to download. Would you please close your eyes because I have to open my Outlook.
It's not going to be much there.
Yeah, well, my stuff is there.
Yeah, no, I sent it to.
Yeah, right, but they sent it to
Then
me.
they sent it to you.
Mm-hmm.
You sure?
I mean, now you're interested, right?
Oh, to him? It's
No.
21 megabytes.
Oh,
Is that about the legal limit?
it might be. Would you mind putting it on a flash drive, please?
Or you can send it. Do you have a Gmail account? Yeah, sorry. I have a big.
You don't have it on a
presentation.
flash drive.
Huh? No, I found it. I'm sorry. Who's on? Is this you?
That's me.
I'll do whatever you want.
I mean, you can start.
I'll do
talking
whatever
and
you
I'll
want.
just-
Oh, use this mic? Yeah, all right, use this one. I'm sorry for the delay. I have a large slide deck, which we'll figure out how to get over. I could take some more slides out. And then. All right, I'll start talking. Okay, thank you. And I noticed a lot of you wearing masks. If you have, I don't know if you read Journal of Clinical Oncology, but the Emory group just showed that patients with myeloma getting active therapy don't make great neutralizing antibodies to COVID-19, despite appropriate vaccinations. So it's very important that we still think about this, especially in patients who are still receiving therapy, because it's still a possibility. You may not end up in the ICU, but you may end up still getting COVID and getting sick from it. My wife has chronic lymphocytic leukemia, so we're always thinking about this. So the other thing while we're at it is there's a drug called Evusheld. I don't know if anybody heard of that. Good. I'm glad to hear. Evusheld is a combination of two monoclonal antibodies, one against something that's more common in COVID-19, the other has finds a different antigen, as you heard about from Dr. Gertz. And so it helps protect you. So it's not a vaccine, but it's the antibodies, it's the end product of a vaccine. We good? Who knows? All right, I'll just keep talking. And so the antibodies have been engineered so that they last in your body for 6 months, as opposed to a normal immunoglobulin, which lasts for about a month. So the nice thing about Evusheld is that it allows you to have a good degree of protection. It's for any patient who has multiple myeloma, they're eligible for it. So I strongly recommend it as something, in addition to vaccinations, you just have to figure out, there we go. You just have to figure out the appropriate timing, because theoretically it might block the vaccinations effort. So I'll start with high-dose melphalan. I'm going to go through a little of everything because melphalan is not the final word. And as you heard from Dr. Gertz, melphalan was first used to gas people during World War I. And if you want to read a really interesting book by Jenna Conant, and she is the daughter of the former president of Harvard undergrad, Harvard University, and she wrote a book about the bombing of Bari Harbor, where mustard gas was released, and it was all covered up by Winston Churchill. It was one of those things. The Germans had mustard gas, the Allies did, and it was kind of like, we won't use it unless the bad guys on the other side do. And so a Liberty ship full of mustard gas was hit and blown up during World War II and released a lot of mustard gas. And again, led to the whole idea, later pioneered by Goodman and Gilman, that nitrogen mustard can treat certain hematologic malignancies, and we still use alkylators to this day, although we'd like to get away from them. This is my disclosures. There's a variety of companies who have done consultancy and honorarius. I thought it's important I let you know that. And I'll be discussing non-EMA, European Medicines Agency, and non-FDA approved indications. So I like this. This is a little vignette from a colleague from the Middle East. Before the new drugs, treating myeloma was like waiting for a taxi and none would come. Then all of a sudden 5 come at once. So I think that's exciting that we have all these options available for patients, but it can be very confusing, and I'll get into why a little bit later on of where do you put all these drugs together. And I know you heard a little bit about that from Dr. Gertz. So this is sort of the history of where we all come from. Now over on the left, and I adapted this from Ken Anderson from 10 years ago, and the slide's gotten a lot bigger because I keep adding drugs and it's really getting crowded on the right-hand side. So I'm gonna have to sort of push everything over. We started in the 50s with Melphalan, and we still have Melphalan. It was used at first orally with a steroid called prednisone. It worked okay, not great. And Dr. Kyles, we mentioned it earlier, has a really cool article on sort of the history of treatment for myeloma, which included orange peel and some other interesting things which didn't work. And then McElwain in the early 80s pioneered the use of high-dose Melphalan. He didn't give a rescue, so these patients were like neutropenic forever as he dose escalated it. And then, as mentioned earlier, Bart Barloghi had a lot to do with the field. He pioneered the use of high-dose dexamethasone, which I know everybody loves getting. But the V here stands for vincristine and erythromycin, which we don't use that much anymore. And then he was the one who really sort of pushed high-dose Melphalan with a rescue, because Melphalan kills good cells and bad cells. It's fairly non-specific. Oh, by the way, BCMA is not specific for myeloma cells. It's also on normal plasma cells, just to correct Dr. Gertz. And as well, CD38 is not only expressed on malignant plasma cells, it's on natural killer cells and a variety of other things. It's very immunomodulatory. We can talk about that later. Anyways, the whole idea of stem cell support came about in the 80s. So this has been around forever. Would I love that we stop doing this? Sure, I'd love to have less toxic things that we can do. So that's our goal for the future. And then, as you know, the IMiDs came out, thalidomide being the first. Horrible reputation for birth defects because it was used for morning sickness. And it was crazy. And there was a woman at the FDA who blocked thalidomide for being used in the US. It was used in Europe and Canada. I forgot her name. Thank you, Frances Kelsey. And Wyeth, who had thalidomide, was pillorying her because why is she not releasing our wonderful drug? And she should have gotten a large medal for that. I think she did. She was given awards because what she did was prevent a huge number of birth defects in the US. However, it was repurposed later for leprosy and then later on for multiple myeloma. That's a whole nother story, which I won't get into now. Then there was lenalidomide, derivative, pomalidomide. And now we have, because for marketing purposes and for patent reasons, derivatives of these drugs are now called the CELMoDs. And iberdomide and mezigdomide, which I haven't yet figured out how to pronounce, are going to be hopefully available in the near future. We have the proteasome inhibitors, as was discussed earlier, and then all monoclonals. Elotuzumab, which is against CS1, also known as SLAM-F5. It's okay, not great, but DARA is much better, as is ESA, both against CD38. But in addition to actually hitting the myeloma cells and killing them, they also do a lot in terms of modulating your immune system. And in fact, some of the CAR T cell programs incorporate potentially daratumumab help deplete natural killer cells? That's another discussion. Some of you may have seen belantamab mafodotin or belamaf. That's an anti-CD38 antibody drug conjugate, very cool. And then sub-Q daratumumab, and then we'll have the bispecific antibodies against BCMA and then GPRC5D, which are teclistamab will probably be approved by the end of the year or early next year, and talquetamab sometime beyond that. And then we have selinexor, panobinostat, which isn't used too often. And then the BCMA-directed cellular therapies, ide-cel and cilta-cel. So we got all these things that potentially will replace transplant, which is fine with me. Just to give you an idea of just how hard it is to take care of cancer patients in general, is that when you go into remission, the level of remission is about 1 billion cells. 1 billion cancer cells is about the size of a sugar cube. So I can take a sugar cube's worth of cancer cells, stick them in me, and I won't find them until I have 10 billion cancer cells. In other words, they have to grow. So it's sort of like the tip of the iceberg phenomena, so that once you go below that tip, you now are treating, but you don't know exactly what's going on because you have to wait and see. So a lot of what we do is based on, well, what do we have for prognostic signals that we get from the myeloma at the get-go? So cytogenetics, stage of disease, a variety of things go into helping us understand what is the prognosis for a particular patient and what's the likelihood the cancer will come back sooner rather than later. That's another discussion, but that's why you get all these tests done when you are first diagnosed to help us understand what the prognosis may be. So what's the best therapy? That's another discussion, which we'll talk about a little bit later. I think Dr. Hillengas will. And so what's the role of melphalan and high-dose melphalan and stem cell transplant? It's really not a transplant, it's a rescue. You're getting your own cells back. A transplant's when you get something from somebody else. And then what's a transplant eligible patient? In France, it used to be up until 65. You hit 65, no transplant for you. But now they're expanding it. In the US, it's really not age, it's fitness. And then everything gets different, fit and frail. So a lot of it's organ function, heart, lungs, musculoskeletal, how much can you walk around? But patients who are in organ failure with kidney failure, who are on dialysis, they can get a stem cell transplant with high-dose melphalan. Bart Paralogi pioneered that. Also, a lot of it's attitudes. Some people don't want to do it. Some people do. And so, I mean, there's all these factors that go into whether or not we should do a transplant. Okay, I'm just going to do some quick definitions. I'm happy to share this. I'll trim it down if you want to put it into a PDF if anyone wants it. How much more time do I have? 10 minutes? I know we start a little late. Just let me, okay, so this is important when you are interpreting data. I know I don't want to get too scientific, but I think it's important because we make decisions on what to do, and the FDA makes decisions on what to do when approving or not approving a drug based on progression-free survival and overall survival. Those are both very important elements when we look at whether or not you're going to approve a drug or not. In fact, you may know melflufen, which was on the market for a very short period of time, seemed to look very promising, but unfortunately, it had a nice progression-free survival, but it had an inferior overall survival. People actually did worse. So, and they're in the middle of trying to salvage that drug. Same thing with venetoclax, inferior overall survival, better progression-free. So very important. Progression-free is the length of time during which the treatment of disease such as cancer, the patient lives with the disease but doesn't get worse, or they've gone into remission and then it comes back. That's progression-free survival. Overall survival is obvious. You are treated for your cancer, and if you're alive, you have met an endpoint, but if you die, you've met another endpoint. So that's overall survival. We want to have, obviously, an improvement in overall survival. And then the median overall survival or median progression-free survival is the time for which, if you take 100 patients requiring therapy, 50 will either have died or 50 will have progressed, and that's the halfway point. So that's very important as well, because you may see all these things in papers or your doctor may describe this to you. So it's good to have a rudimentary understanding. I don't understand all of it either. I have to talk to our statisticians half the time. So a Kaplan-Meier analysis, I'll go through this very quickly, is to estimate progression or survival. And then you follow all patients, but everybody doesn't get treated on the same day. They're all treated on different days. And then some people may say, I don't want to do this anymore. So they go off study, or they move out of town and nobody can find them. So there are all these things that happen while you're trying to do an analysis. These are all the things that make cancer treatment complicated when you're doing a study, because you got to make sure that you're accounting for all of them, and that also they don't have a toxicity that you're not expecting, and that has happened with several drugs. And so this is I got this it's a Vanderbilt thing from WikiPub. And so these are people who've all gotten treatment and representation. This is just them all lined up, and this is time. And so different things happen to them. Some of them may die, some of them may go off study, some of them progress, and they're all starting at different time points. So what a Kaplan-Meier curve does, it lines everybody up as if they're all starting at the same time. So it's very important. Now, if you've heard this before, I'm sorry, I'll go through it quick. So this is a very simplified Kaplan-Meier curve, just showing percent of people surviving, but what happens is things may happen. They are censored. They leave town, so you can't follow them anymore. So the curve drops, and then some may meet an endpoint. They've progressed or they've died, and so the curve keeps dropping. So this gives you an idea of how something is working relative to something else, and I'll show you that in a little bit. So this is from a fellow named David Robinson. He presented in Blood few years ago. And what it shows is, actually, this is an abstract. He presented in a different journal. And what it shows is how we've gotten better over time in terms of patient survival. You can see here back in the 70s to 1980s, the median survival here was about, it was pretty bad. It was about 3+ years. So that was back in the day when we really only had oral melphalan. And then as you see over time, we got better so that the median survival keeps rising. So here, the 5-year median survival for patients who were treated, the median survival for patients treated from 2001 to 2010 is about 5 years. But I'll tell you how that's even gotten better. And this is another one that was an abstract, which they published in another economics journal, I think. And this just shows how things have improved. The blue line here is for people less than 65. And then from 2006 to 2010, and this is for less than 65 from 2011 to 2012. And you can see on the Kaplan-Meier curve, things have gotten better. But what's really cool is for the over 65 crowd from 2006 to 10 here has now improved to up here. And you can see it stops here because you have to have patients live long enough for you to be able to follow the curve. So that's another issue. But this is exciting. We've gotten better at what we do. And then lastly, here's auto transplant. It's very simplified. There's our patient in green. This patient has their stem cells collected. Many of you who've had a stem cell transplant know what it's about. We used to do bone marrow harvests, which are pretty crude. Now we do peripheral blood, and even then it'll change because maybe it'll always just be CAR T. Cells are put in the freezer and then very cold. And then patient receives high dose therapy. And people have tried to improve upon melphalan, and really it's still melphalan. So that wipes out the hematopoietic system, cells are infused back in, and then patient is rescued. And for those of you who've had this, you know it's not a lot of fun. So I'm going to start to wrap up. This is the determination trial. Paul Richardson led this. It was just published a few months ago. And this was RVD, which at the time was the gold standard. Everybody got RVD. And they not only did it in the US, they did it in France. And we can talk about it later, the difference between the French trial and the American trial, because there are differences on the maintenance arm. And the patients who got RVD only, got it for 2 or 3 cycles, had their stem cells collected, and then got RVD for up to 8 cycles. And then the transplant arm got RVD induction 2 to 3, stem cell collection, high dose melphalan. Then the back end, they consolidated. And this is very popular in Europe. In the US, you've just had high dose melphalan, it's day 60. Oh, we're going to give you more RVD again, more patients. Are you kidding me? So, that's why we don't do that much. And that's another study where we showed that it probably doesn't matter if you've gotten an RVD induction. And then they all got Len maintenance. And the primary endpoint was progression-free survival. And then there are other safety endpoints and overall survival. And this is that Kaplan-Meier curve. For the patients who got a transplant, they had a superior progression-free survival. In other words, it benefited them in terms of when the disease came back, it progressed, or they were in remission and they had relapse. And the blue line are the patients who got RVD only. And so the median progression-free survival, remember, that's why I gave you all that stuff at the beginning, is 46 months for RVD alone, and it's 67 months for those who got a stem cell transplant. You say, well, why doesn't everybody get a stem cell transplant? Well, the overall survival is not that different. Why? Because we got great salvage options for patients after progression from if they have a stem cell transplant. So right now, there's no difference, and we don't expect there will be over time. So I'm going to end here with a few other things. People have tried to improve on things after transplant. This is one using elotuzumab as part of consolidation maintenance. This is a German study. And you can see here, they had all these different permutations. Lendex alone, Lendex plus Elo, Lendex alone, Lendex plus Elo. And they had different approaches. You could either get a 2 transplants versus 1. But anyways, the bottom line, no difference when you added elotuzumab. Now, the French study added, this is VTD, which is bortezomib, thalidomide, dex, transplant, and then with or without DARA, and then DARA versus observation. And this shows that DARA as a single agent compared to nothing benefits people with, who received the daratumumab as opposed to nothing. As you know, now the gold standard is, or at least the FDA-approved gold standard is lenalidomide. We can talk about that if you wish. But this is where it gets really confusing. It turns out that the patients who got VTD, thalidomide, bortezomib, and dex, and then no maintenance, they are here in terms of their progression-free survival, inferior. Here, if you look at it though, the patients who got VTD DARA and no for just alone, they did very well. But those who got VTD DARA and then got DARA on the back end for maintenance, they're about the same. And here are the ones who got DARA, VTD, and then observation, also about the same. So daratumumab, as part of induction, seems to have a halo effect that goes well beyond the initial, whoops. Was that me, or are you trying to tell me to go to the next slide? No. It's an interesting phenomena. So DARA, in addition to killing plasma cells, modulates the immune system. We're just beginning to understand how that works. I can talk about that a little bit later on. So I'm going to end this part of my talk with what the Princess Bride teaches us about outcomes in multiple myeloma. And the sad part is, when I say this to students, they all go, "What's the Princess Bride?" And I'm like, oh my God. So kids, I guess it's like when you talk about the Rolling Stones and everybody looks at you, oh, those are a bunch of old men. So I always do recommend for those of you who have not seen The Princess Bride to go see it, because where Vizzini and his, well, as you know, he kept saying inconceivable when something he thought could never happen happens. So his last word was close to inconceivable when he was having with the Dread Pirate Roberts, when he's having the game as to who's going to drink the poison. And so I know Kane poison. So Inigo Montoya is played by Mandy Patinkin, and Vizzini's played by Wally Shawn. So he goes, "It's inconceivable," says Vizzini. And having heard this claim 3 times only to be confronted by alternate realities, Inigo Montoya says, "I don't think you know what that word means." So how, you may ask, does this relate to multiple myeloma? 20 years ago, it was inconceivable that the median overall survival for myeloma patient would now be beyond 10 years. In the 1980s, it was 3 years. Now we're in excess of 10 years, with or without transplant. And to consider alkylator-free regimens for fit or failed patients has become all but routine to use induction regimens that don't use alkylators. Inconceivable. And that post-relapse outcomes could nearly be as long as the initial responses, if not longer in some cases. And the results of phase 3 trials could be confounded by post-transplant therapies. Great problem for the patients. hard problem for the investigators, because it makes it hard when patients are getting great salvage therapies to say, well, we're going to use overall survival as an endpoint, because now we're able to treat patients quite well. In fact, sometimes even better than the induction regimens they received. Some of the older studies, we used to use a doublet, Lendex. That's like crazy. And so if you get a triplet, so you get Dara Lendex or Dara Pomdex, you may even have a longer response than the original Lendex. So it makes it very difficult to figure out what's going on, even in the context of B-cell maturation, antigen-directed therapy. So I'll stop there. I can talk about CAR T cells. I have some cute slides on that a little bit later. Thank you. That's not mine. Oh, there it is. Okay. I will just close it or leave it open?
Yes,
Close it?
they can take care of it.
Sounds good.
Sounds good. So this is, for those of you who are of my generation and remember Paul Harvey and the rest, now you know the rest of the story. So when Dr. McCarthy was asking me about time, what I failed to tell him was that I left my reading glasses and my phone up here earlier, so I had no clue what time it was. And then to make it even worse for our online audience, what you don't see is we have two clocks here that have times that don't reflect accurate times. So we are all, and both of those clocks have different times, so we're really confused. Jens Hillengass, I have known Jens now for close to 16, 17 years, I believe. Jens would not be on the Mount Rushmore of myeloma. And the only reason is because he's not as old and crusty as Dr. Gertz and Dr. McCarthy. And I can only say that because I like them. And I think Dr. Hillengass is really one of the brightest minds in myeloma today. And you are so fortunate to have him here at Roswell Park. I knew him when he was in Heidelberg. And when I found out he was coming here, I was just ecstatic. I thought it was just absolutely wonderful. So with that, I'd like to introduce Dr. Hillengass, and then we'll break after that.
Thanks for the great introduction, Greg. It's always challenging to talk about these people like Dr. Gertz and Dr. McCarthy, who are in the business for such a long time and have so much knowledge, not only because of experience, just because their brains are much bigger than mine. Where are my slides now? They were there, I know it. Sorry, please close your eyes again. I don't know, protected view. I'm in the view. I'm on the drive. I apologize. It is actually there. There we go. So there were great introductions to what I want to talk about and what Greg asked me to talk about. Diagnosis, and I don't want to, sorry, I walk away from the microphone. I shouldn't do that. It's a never-ending story, which is a good thing in this case. We are in development, and you heard the very early steps from Dr. Gertz. You heard the further development from Dr. McCarthy, and I can just piggyback on that. I work with some pharma companies to do our clinical trials and to learn more about the disease because they have the money, and they're very supportive. So I'm really grateful for that. The topics that Greg gave me, sorry, I take my mask off. The topics that Greg gave me are 3 to 5 years, and I will explain where this is coming from: a changing perception, diagnosis to determine strategies, and diagnosis a continuous process in the area of novel therapies. 3 to 5 years, that's actually when I started treating myeloma. And I'm in that age already, even though I'm a little bit younger than the speakers before me. But when I started treating myeloma, it was 20 years ago. So I do look a little bit younger, people tell me, than I actually am. So I really treat myeloma for almost 20 years now. And at that time, Salt Lake City, which was mentioned, where HealthTree is, was where the Winter Olympics took place. If you remember, most people in this room do, I guess. American Idol aired for the first time. Can you imagine there was a time before American Idol? And Pierce Brosnan was still James Bond. So it's that long ago that I started treating multiple myeloma. I was surprised myself when I Googled it, what happened in the year 2002. The standard of care for myeloma back in Germany, and I think to a certain degree even in the US, was VAD, which is something we don't even use anymore because we know it's not really good. It's alkylating agents, agents that are destroying DNA, so the things that Dr. Gertz explained earlier, that was the only thing we had. And then these alkylating agents that we mentioned, the melphalan, for patients who could not get a stem cell transplant with a lot of melphalan, they got just less melphalan, and that's what we did, right? And that's actually the time when I came into multiple myeloma, and the survival was fairly short. I mean, we didn't talk about quality of life. Patients wanted to live a year longer, maybe, right? So that was the topic that we were talking about at patient meetings at that time. And we waited to treat multiple myeloma patients until they had so-called end organ damage, meaning they had holes in their bones, they had kidney failure. Unfortunately, that still happens because we don't screen for myeloma, and most of you are diagnosed because you have symptoms. But in patients who have an early stage, we obviously treat earlier nowadays. The reason for that, and those were the criteria, so the famous, infamous CREB criteria: high calcium, renal insufficiency, anemia, low counts in your blood, or bone lesions. So those were the CREB criteria. That was the reason to treat. Why did we not treat earlier? Sorry, why did we not treat earlier? And a lot of people from breast oncology, colon cancer, they say, we don't wait until my patient has metastases in their bones. I treat them earlier. But they tried that, and they used the drugs that you heard about, these alkylating agents, this thalidomide, and it was just not better. The patients had the myeloma later, but they didn't live longer because they just got it later, got symptoms and side effects from the treatments earlier, but their survival, as Dr. McCarthy explained, the overall survival was not better. So we said, let's wait until we really have to treat, and then we treat, because then we can kind of justify that we give our patients so many side effects. And this, you already saw the time that the patients lived, and it got better over time because that's the area where thalidomide was introduced. So it got better, but it was still not really exciting. Today, the diagnosis, and I don't want to go into many details with what's going on with genetics when we look into the tumor cells to look what genes are upregulated, downregulated, what chromosomes are broken or moved from one part to the other. I just want to kind of give you the big picture. Why is it important to know as much as possible about the myeloma? Because we now make our decisions based on that. And the first decision is we added certain prognostic markers for patients who have early stages on top of the CREB criteria. We don't want to wait anymore until the disease progresses or until the disease really has harmed the organism, if we know about it, obviously in early stages like smoldering myeloma and MGUS. And now we call it the SLIM CREB criteria. And you can Google that and find it easily what the SLIM CREB criteria are. So we treat earlier. That's the first approach, because the treatments that we have are still a lot of side effects, but much less toxic than initially. And they have shown that if we treat earlier, we have a better outcome for our patients. Another thing that was in 2002, when I started treating myeloma, we had not many tools. And you know this, the saying, and I learned, and Greg is teaching me idioms, American idioms. And I think if one of them is, and Dr. McCarthy uses it a lot, if you only have a hammer, everything looks like a nail, right? And that's actually what we did. And you can see on the right side, I think those targets are not really suited for a hammer. So that's what we did. We treated every myeloma patient with melphalan because there was nothing else, right? And so nowadays we have a much better problem because we have many more tools. Now the challenge is to find the right tool for the right patient. That is what we do with our diagnostics. We do the bone marrow biopsies that everyone loves. And Dr. McCarthy and myself just had a few months ago, both one for our research to learn more what is the healthy bone marrow. And sorry, that was us. And compare that to our patients and see, okay, what are the differences? Where can we fit a tool that fits to the target that we are treating? And venetoclax was already mentioned. It's not approved for multiple myeloma yet, but it's one approach that we have to treat patients with something that fits to their disease. And that's something, then the next question is, when do we do that, right? The sequencing, should we do that right away? Because we know, so this study showed that this venetoclax, a drug that is used in other cancers, where we thought, hey, that should work in myeloma as well. And they tried, and you see on the left-hand side, this PFS, the progression-free survival that Dr. McCarthy explained, the time until the disease came back, was longer with the venetoclax for the patients who got it compared to, together with the bortezomib compared to a placebo. But then they looked at the overall survival and can see the red curve is higher. So not getting venetoclax was not statistically, but it was better for the patient not to get this new drug. That's not how this works, right? So why is the progression-free survival better, but the overall survival actually worse? And that was the reason we have to think, why do some patients benefit and others don't? And so what they found, there's a change in the cancer cells, in the myeloma cells. One is called a translocation 1114, where when the cells divide, a small piece of the chromosome 11 is transferred to the chromosome 14, I think, or vice versa. So they're on the wrong part, because that happens in cancer patients. They cannot repair themselves so easily. And if you have this translocation as a patient, then those patients, you see the blue curve is much higher. And on the right side, there's another genetic change. It's called a BCL2 mutation, or BCL2 is upregulated in those cells, and those patients benefit as well. So we finally come to a point where we can adjust our treatment to patients. Patients who don't have these changes, I would not give this treatment, because as you have seen, it's worse. In patients who do have the treatment, these changes, there we can give it. And that's only one example that we adjust. And we are always eager at our meetings when someone is presenting something, saying, okay, in this subgroup of patient, it works better. And that's what we are looking for. We don't want to use this one size fits all approach anymore. We still do it in a lot of settings, but we want to be more specific for patient, and we identify more targets that can be targeted with a certain treatment. So, and the diagnosis continues. It's not only at first diagnosis, because what we also know, the myeloma cells mutate. And so they are not, if you do a biopsy at first diagnosis, a bone marrow biopsy, and you do it after the first treatment, after the second treatment, after the third, the cells look a little bit different. Not always and not all of them, but we know there's something that we call a clonal tiding. So some clones are more susceptible to one treatment combination, and another clone set is more susceptible to another treatment. And unfortunately, we are not that good yet that we know, okay, this will be the best treatment for this patient. So we have to try out, unfortunately. So we see, for example, if these 5 classes, and there are even more to come, Dr. Gertz talked about, if you have a certain class or combination of drug classes and give that to a patient, and you see it works well, you can, as a doctor, consider giving something like this or something similar, maybe another antibody, but combined again with an immunomodulatory drug, or combined with a proteasome inhibitor later on when the disease comes back, because we kill initially certain subgroup of cells, and then it comes back. It's a more kind of different subgroup of cells, and that's why it's called multiple myeloma. Some people call it even multiple myeloma, because it's really developing and kind of developing defense strategies against our treatments. And then we treat with a different combination, and then we treat with a different combination. And that's what we call the novel therapies. We don't use the one size fits all anymore. And what we are also doing, we look for the response. So if we see that a patient has a really good response to a certain treatment, we know that's good. So not a lot of cancer in your body is a good thing. And you might be very surprised to hear that, but that's what I can tell you. Less cancer in your body is better. And that's what we do with the so-called MRD. And you might have heard about it. It's so-called minimal residual disease. And that's something, or measurable residual disease, because we can only measure to a certain level. And unfortunately, even if you're MRD negative, you're not cured from the myeloma. It's just we cannot measure it anymore, but at least it means it's very, very low. Nowadays, we have technology that can measure 1 in a million cells and sometimes even better with that sensitivity in the bone marrow. A challenge is that if we do this, so it's good to have this MRD negativity. If you're MRD positive, we can still treat, but there are other things that we have to consider. But one challenge is that we do our bone marrow biopsies, as you know, always here on the pelvis. And if we do a PET CT, for example, you know, it's this way it lights up like a Christmas tree, unfortunately, sometimes. And we can identify areas where the myeloma is growing. If it's growing in the pelvis and you do biopsy there, then we know what's going on. But if it's growing somewhere else, then we have a challenge, right? We know that there's a pretty good correlation, but we also know that you can have all of the above. You can have a PET positive, MRD negative patient, you can have a MRD positive, PET negative patient, and so on, right? So you can have all these combinations. That's why we do our diagnostics later on with bone marrow biopsy, blood work, obviously, because it's the easiest, and imaging, right? So we combine all these things to get a whole picture of our patient. In summary, our treatments have improved. I hope that's something that you remember from all 3 talks, and there will be more about that later. They still have to become more individualized because, as I said, some patients respond very well to one set of drugs, while they don't respond very well to another set of drugs. What is even another topic, which patients develop which side effects to which drug. And we have done back in Germany, we have done a large study where we looked into the myeloma cells and the microenvironment of certain patients and looked, okay, do they develop this polyneuropathy or the peripheral neuropathy that most of you unfortunately know. And we couldn't find anything to identify patients beforehand because we would not give them treatment like thalidomide or bortezomib that can cause this neuropathy. And some patients do great with it and others have really, really struggles. And then response assessment is becoming more sensitive. Back in the day, we could maybe find 1 in 100 cells. Now we can find 1 in a million cells or even less. And that is something that guides us in our treatment. We know when the patient is in a deep remission, and then we can go on with and adjust our treatment according to that. Thank you very much.
Thank you. Well, that was a wonderful start to the day. For our online audience, we're going to be back. We're going to leave you now, and we will be back in 15 or so minutes. So we'll sign off to you now. And for those of you in the audience, we have, I don't have many, but first come, first serve. We have these new T-shirts that we just brought out for the Northeast Myeloma Community Chapter with the sponsors of it on the back. I have 2 large ones. We also have a bunch of mediums, extra larges, and a few double extra larges. So when you come on out during the course of the day, if you want some T-shirts, just take one as you see them out there. And we will be back in 15 minutes. So we will be back here at 10:35. Don't pay attention to the clocks here. So, and I'll start yelling at you about 3 minutes before we convene again. And remember, we're in the room next door. If you want to get some water or refreshments or stuff, we still have things there. Does anybody? I have 2 large T-shirts. Anybody want to call dibs on the large?
Sharing class in her last so that we don't have to roll the thing up because you
Well,
cannot turn off the.
Oh, you can't turn it
projector.
off?
No,
Okay.
that's the problem. I wanted to mute it.
Yeah, that'd be better than the
Oh, but
sponsor. I'd like to keep this one, but it'd be better than the sponsors.
volume, no.
This doesn't seem to be
But
working.
we can.
Does this work?
Did you turn it on?
I don't know where.
Push it on the top here.
Oh,
This is mute. This is no mic.
yeah, I can't see a switch. I mean, there's. Oh.
It's mute.
Or if we can get one more mic.
Yeah.
Because I have one there and
For
I
the
would need
questions,
one
yeah.
for me to ask the questions. Strange. Oh. We have the screen up, but I don't know if the audio. Well, we were deciding right now, can we do the HealthTree logo behind that? Yeah. Yeah. And would that interfere with the lighting? No, it's because it's a nice green instead of real bright white. Okay. This, do you know how to turn this on? Yes. Oh, he knows. Oh. We were looking all over. This is on now? No. How do you turn it on when because I'm gonna use it for them and then they're gonna pass that. Just hold it down for a second. Yep. Okay, turn it back off. Just leave it open. Okay, which time? And I said, what time? We got to 10:35. So I'd say 10:25. So we'll say.
I was just curious for the, you need to ask questions.
Oh, oh. No, I'll just have them use this.
Yeah, that's what I was thinking. Action.
There's another one. Okay, I guess, oh wait, how do I, is this on?
It is on. I just muted it. Do you want, are you ready to go?
No.
Okay.
Oh, you're muting.
Yes, I just
Fabulous.
muted it.
Yeah, you can put, I'm gonna go round them all up. Okay, I'm gonna round them up and then we'll get going. Not yet. Good. So basically, we're going to have a conversation with everybody, and then that will morph into the audience Q&A, and then I'll get
Okay.
up and do that.
If you want, I have about 5 slides on part T if you want me to talk about it or not. It's up to you.
It's up, well, if, let's ask if here they are. If we ask you guys to pull up a slide, could you do that during the course of the talk? Or is, yeah,
They could, yeah.
okay.
It's in my
Can-
slide deck.
Could you point out,
where
and
I
then what
left
we can
off.
do is where you left off.
With the Princess Bride.
And then when you we come to you, and if you want to, you
Yeah.
can just ask, can you put on the slide
Sure,
and blah, blah,
sure,
blah.
And then we can flip through. Because
So we're going to
that's
start, since it's abbreviated, it's going to be, the beginning is going to be all about CAR T, then we're going to go into some general topics.
Well,
Then
yeah,
Then I'm going to get up and do the Q&A.
Yeah.
And then we'll go to lunch.
'cause I have about 5 or 6, I can zip through them, but
Okay.
the whole point is that CAR Ts is not the final word.
Right.
That's
See,
very important.
I really like improvisation, so that's why I don't script these things or
Yeah,
anything.
yeah,
I want
no
you,
worries.
because you don't need to be told what to say.
Yeah, no worries.
Okay, are we all here? Let me go round up, make sure we have any stragglers. Excuse me.
Interesting. Yeah.
Okay, we're about to get started. You, wow. This were a California crowd, man, it would take them forever. Okay, we're gonna get started. Okay, if we could have our speakers take their seats.
And we hand it up.
Jaden, can you unmute me?
Oh, yeah.
And then that mic is for you to pass around. Any order? And Dr. Oh, there you are. Is that me? Or was that? Help me, Jaden. Or
Is it?
let's just turn it off.
Let me mute it. Let me take it.
Yeah, I think
You can pass that around, yeah.
But-
Let's see if this works, so let me see if I fixed it. Hey, I fixed it, I think.
hang on, text. I don't think I'll clip it. I'll just.
Yeah. Careful with that.
Let's do that. I am not the most technically proficient person in the world. So one of the things that I like to do with these meetings is I like a lot of improvisation and I like a lot of repetition, because I think for lay audiences, it's really important to hear the same concepts many times during the course of the day, and then it will set in. I'm a former middle school teacher, so that's why I think that way. So I'd like to start out today talking about CAR T, because that's what's on top of everybody's mind. But there are obstacles with CAR T. So I'll start with a real simple one. Who should be excited about CAR T? And we can start with anybody, and I hope that you will all add and pass the mic along.
Since I have the microphone in my hand and it's a simple question, I will take that one. No, I mean, we were very excited when it came, because as all drugs and all treatments that we use, all new ones, are tested in patients who have very advanced disease. And because there it sounds mean, but oftentimes you have nothing to lose, right? It's worth the try to find something that still works if everything else has failed. And usually we see response rates that are, well, okay, there's some patients have a benefit, most don't. And in the CAR T cells, it looked like it's almost the opposite. Most patients had a benefit in those very late lines. What we have learned now, as with all other treatments and drugs that we have as well, the earlier we use, it seems to be the better. And we're still in early stages, and obviously here are people who are much more proficient in this area. But I think one exciting thing is that it even works if the disease is really not responding to a lot of other things anymore. That's one thing that patients who are basically didn't have a lot of hope have hope again. And another is that we might use it earlier, which then benefits everyone, because we can use it in areas where the patients are still in much better condition. They feel better. They have had less side effects over the course of their treatments. And I think those are the areas. And
I am not the moderator of these things normally, and I just Jenny was very ill and did end up leaving earlier. And the other thing I didn't do, which I realize, is I didn't introduce our fourth speaker, who is Dr. Urvi Shah from Memorial Sloan Kettering, and she'll be speaking this afternoon. So I asked her to join us for this discussion. I would never make it on the Oprah show because I don't understand protocol. So I will pass that back to you now. Thank you. Does anybody have to add to that? So who should be excited about CAR T?
My turn?
Yeah.
All right. You put up the slides. If you want, I can go if while you're waiting.
Just to add, I think, when we ask who should be excited just now, I see patients with newly diagnosed myeloma or early stage getting excited about CAR T, but I'd say pause on that. Only patients with 4 or more lines of therapy it's approved. And, CAR T, while it does have great response rates, there are situations in which it doesn't work. It takes time, to prep the patient for it. And there are other good therapies. So we have to do it in everything in a sequence, and the CAR T is not the right thing for people initially, and it's only in certain situations later. So while it's exciting, it's not for everyone and not in the beginning. And I think that's quite important for patients to know, because many times patients hear about this like miracle treatment, but it's not miracle for everyone, and we have to find the right patients that it works for. It also takes some time to make the CAR T cells. So if the disease is very active, then that's not a good patient also to try to do CAR T cells for.
Can we get the slides for?
Thank you. What, you're not talking? I can't believe it. You have nothing to say? Come on. It's like a first. I can just put in, careful what you wish for, because CAR T, at least in lymphoma, we have a longer track record. And I have some patients who are 4+ years out who still get GCSF twice a week because they don't have good blood counts. Now that's a CD19 directed CAR, that's against B cells. But why should your knocking off your B cells influence your neutrophils? We don't understand that. They also, this person, she's got a low platelet count, and yet she's got a cellular marrow. Her platelets are in the 30s. So when she had to have her hip replaced, she had to be tanked up with platelets. She's grateful. Her lymphoma is gone. We're not quite seeing a long enough term follow-up with myeloma patients, so we don't know. Plus, as you know, the waiting list is ridiculous. So I've had patients die while waiting to get a CAR T cell therapy. It's horrible. And so we struggle with that. Both BMS and Janssen are struggling with manufacturing. So it's CAR T 101 right now. And so we really have a ways to go.
Well, what are some of the obstacles that you're talking about? Can you explain those in greater detail?
Waiting
So like
list.
what are the rules that you have to adhere to?
Waiting
Who can
list.
get them?
Waiting list.
So how many CAR T procedures are there done generally in a month in this country?
I have no clue, but it's not a lot. I mean, we have how many?
What do you mean? How many patients? A waiting list, we have a long wait list,
Yeah,
and
we
we
have at
would
least
like to
10
do it
patients.
at least.
And our next slot is November. So that's a huge problem. I mean, we're just opening Janssen product and we're going to be sharing it with the University of Rochester because they don't have it either. So, and it's just that the manufacturing has taken forever. Mostly because I don't think companies realize this isn't a drug, it's a living thing. And when it's a living thing, people are variable. So somebody's immune system's beat up, you may not get good lymphocytes out. And so right now we're trying to understand why that is. The Penn group had a really interesting study where they grabbed CAR T cells like right away. The patient had high risk features, and they found that the type of lymphocytes they got then were much better than you waited for a patient to relapse refractory to 4 prior lines of therapy, including an IMiD, a proteasome inhibitor, and a monoclonal antibody to CD38. Those are patients whose immune system's fairly beat up. So now the whole issue is, well, maybe we should store lymphocytes on somebody who's high risk. So therefore, you've got a much better set of lymphocytes to manufacture. However, some of the manufacturing can only be done on live, fresh cells as opposed to frozen. So there's a lot we have to learn to make this better and also understand better so that we don't treat everybody with a CAR T cell.
And that brings us to the alternatives to CAR T and bispecifics. How do you see bispecifics coexisting with stem cell transplant, coexisting with CAR T?
Yeah, I think that the reality of CAR T in its current iteration today doesn't live up to the hype. There are real issues with supply chain as well as technical expertise and employment levels at the manufacturing facilities. The bispecifics, of which the first has already been approved in the European Union and anticipate approval in the United States, will be an off-the-shelf, something that'll be amenable to repeated administration as opposed to CAR T, which is a one-time and done. And given the very high response rate with bispecifics, I think it will be very, very powerful competition for CAR T, where I would anticipate no wait lists for bispecific antibodies and the ability to continue treatment long-term, which is not generally the case for CAR T. So I think it's actually more exciting than CAR T. Nothing's perfect, and the bispecific antibodies have been associated with some extremely, extremely serious infections. And that's part of the learning curve, unfortunately, is to just understand what it does to the normal healthy immune system and what can be done to prevent these exotic infections. This business with CAR T, everybody talks about effectiveness, but some of the side effects can be quite sobering. With one of the products, there have been reports of late, after 6 months, Parkinson's disease. I've never met a patient who's been told about the side effect. It definitely occurs, and regrettably, CAR T has a mortality rate. I mean, people don't always survive CAR T treatment, and I would say that mortality rate, at least at our institution, is between 5 and 10 times the mortality rate of autologous stem cell transplantation. I think the exciting thing, Greg asks about, what about getting excited about CAR T? I think that Professor McCarthy was correct when he said it's CAR T 101, and that in its current iteration, there's some serious obstacles. And it's not that great, but the opportunities for further development, different targets, multiple targets, CAR, not T, but CAR with other cells, suggest that there's a very high research upside to try and develop better products than what we currently have, which are highly effective, but not the magic bullet yet.
Will the patients in this room ever see the day where stem cell transplants become obsolete?
Maybe.
Why maybe? What is the thing that what makes it a maybe?
Well, it still works. And it depends. We're in the US and we're very lucky. We have access to a whole bunch of things that people outside the US don't have. So even in Europe, there are high-dose malformed stem cell rescue is part of the treatment continuum for a lot of patients. And a lot of other drugs are not available yet and won't be for a while. And that's just because of the way medicine works in other countries. I'm not going to say our system is great, it's chaotic. There's a whole bunch of things we could spend an hour on what's wrong with healthcare here, but we do have great access to drugs. It's just all financially unsustainable. That's another issue. But yeah, I do think, and then high-dose malformed stem cell rescues fairly cost effective as opposed to a quad, which is going to be what we're doing next. We're going to go to a 4-drug regimen. And so I do see the day that potentially will go away, but we also have to think of what can we do to replace it? Will it be a CAR T cell? But we may want to collect stem cells so that we can rescue people who have pancytopenia after the CAR T cell therapy, which occurs. So there's a lot we don't understand. And as was mentioned earlier, Janssen is studying it. We already know that ide-cel will likely be moved up because one of their trials looking at salvage, earlier salvage with dealer's choice versus CAR T is going to be, it's a positive study. So we'll be using it earlier, but then if somebody has beat up cells and you can't get any drugs into them because they have low-grade pancytopenia, we better collect everybody up front. So we'll still be collecting stem cells. It's just that we may not be using them with high-dose malformed. We'll be using them with something else.
So stem cell transplant is 40 years and running in multiple myeloma, and to date hasn't been knocked out of that spot. Most investigators report a fraction of patients who are 10 and 20-year survivors after stem cell transplant without any maintenance treatment. And it works a completely different way than all of the other available treatments. As things go along, the biggest concern that I have in my practice is the fact that patients who get stem cell transplant do have a higher risk of second cancers. And as people live longer and longer and longer, they have time to develop these problems. And when they develop, they're quite unpleasant. And so it's, for me, kind of the big problem in the back of my mind about, will transplant lead an otherwise long-term good outcome patient to develop a second cancer? But for right now, I think it would take quite an impressive outcome to dislodge stem cell transplant from our current practice.
Yeah, just as a follow, we've had patients who didn't get a transplant, but then got lenalidomide-based RBD and have developed acute lymphoblastic leukemia. We don't understand that. We have no clue how that occurs and why it occurs in a fraction of patients. We also don't understand. We also don't understand there's a paper from Olangren, about 10, 15 years ago, looking at the Swedish Cancer Registry And what he showed is in that patients who had MGUS, monoclonal gammopathy of undetermined significance, Dr. Kyle's observation, and smoldering myeloma, who hadn't been treated yet, still had a 2 to 3-fold increased risk of getting AML, acute myeloid leukemia, and MDS without therapy. So myeloma itself predisposes people to developing hematologic disorders. We just don't understand how this works. Then, of course, you pile on drugs on top of it, the risk starts to go up. But as to Dr. Gertz's point, it's better to be alive to be at risk for developing a second cancer than not to be alive, and you won't get a second cancer. So we just need to do a better job of identifying those patients who we should treat differently because they've got a different genomic makeup than other patients who don't develop the second cancers. Oh, and by the way, there is are you 19 or 20 years from your stem cell transplant in the audience? Where are you? Well, anyways, there is one of our patients is here who has transplant about 20 years ago. No maintenance, doing well. And I don't understand what's different about him relative to other patients. So we need to understand that a lot more.
Two weeks ago today, we had a roundtable in Hamburg, Germany, and we had a session with a panelist who was the first CAR T patient that this doctor had treated, I guess 3.5, 4 years ago. Hasn't been on any treatment since, and he's just doing fine. Not that he's the typical case, but there are cases like that out there, and it's a really exciting to see them. And he had been through 10 lines of therapy. And has been without therapy now for going on to 4 years and is feeling just wonderful. What do you see as the future? I mean, first of all, I guess we have 2 approved CAR T cell products right now. What do they presage for the future? I mean, one looked at 1 target, one goes after 2 targets. What is the future looking like for CAR T? How is it going to change? Is it going to stick with figuring out how to exploit BCMA, or is it going to be other things that are going to be coming up that are in the research right now?
So we already have other targets being looked at, right? So the GPRC5D CAR T cell is there, and similarly, you have a GPRC5D bispecific. So we have that trial open at our center with the CAR T cell for MSK, but there is a multi-center study also ongoing with that. So we already have that target. Then the next one is cevostamab or FCRH5. So there are different targets being looked at. I don't know what's going to be next, but there are many different targets being looked at. So I think, how do we combine these targets? Do we do them individually? The way some of the thoughts are also, can you put 2 targets on the same CAR T? Do you have 2 different CAR T products and you give them together? Should we give them earlier lines of therapy, or should we give them later because they have more toxicity? And I think each CAR T also has different side effects depending on what they're targeting, right? Like, so if it's BCMA, then there might be a little bit more immune suppression compared to if there's GPRC5D, which may have skin and nail side effects. So there's a lot of difference and individualization that's going to come. And I think we have a lot to learn from the bispecifics too, because once we start using the bispecifics, if that's going to be much easier to give. And I think the challenge may be a little bit more like the risk of infections long term. And so once we understand how that's working out in the real world, I think we'll be able to better understand. And the last thing I'll say is just access to CAR T. Just now, the companies haven't figured out how to make enough of it. So if that's not, done over time, then we'll just be using bispecifics because of that. So I think there's lots still to be determined, and the next few years will tell us.
And I wanted to piggyback on what was already said. We need better immune system. We need better T cells in a patient. There are attempts to get T cells from healthy donors or other kind of worms with challenges, because those T cells think, oh, wow, I'm in the wrong environment. I have to fight everything. But there are approaches like that so that the T cells are not harmed by the myeloma, as Dr. McCarthy said, or harmed by the treatments that we have done. And I think approaches to make those T cells stronger and better and longer living, so that they don't die after a few months, but stay in the body longer and fight the myeloma longer, which is then more similar to the bispecific that we can give over months and hopefully years. I think that's another part. and then figuring out the side effects, why are they happening and what can we do to prevent them. I think those are future developments.
We're going to think of your questions. I'm going to come out and get your questions right after this, and we'll close this part with this question. My background is in politics and history. So as you can see, this morning was a lot of historical stuff. What's the one issue or one problem you wish we could overcome soon? I mean, what's the one thing that you think is within grasp that we don't have yet? Or what do you see in the future that people should get?
You always have something to say. You got to think, all right.
Start with Dr. McCarthy.
Medicine-wise or economically?
Both. Any issue that you think these people should think about in the next year or two as they think of their therapy?
Well, they shouldn't have to think of the economics, okay? That's ours and politicians and insurance companies and all that. Although I do recommend there's a video by Katie Porter, and she just grills the head of makes venetoclax, AbbVie. And she gets out her little whiteboard and she talks about, and she goes through the fact that most pharmaceutical companies spend more money on advertising and taking out physicians to dinner than they do on R&D, even though they'll say otherwise. Oh, we have to have these large, I'm going to get very political, sorry. But so this is why you need to tell your Congress people and senators and everything else, look, we have to have a different approach to pharmaceuticals because the cost is ridiculous. She does the same thing to Mark Ellis, who was then CEO of Celgene. Oh, the price of lenalidomide went up 5 times. Did it get any better? Was it more effective? And it still is that way. So that's number one. We have to have a different approach. This is a global across the board. We have to look at the cost of medications. And even when Len becomes generic, it's still going to be ridiculously expensive because we have to have the REMS program, which you're all familiar with. And I always love it. People in their 70s have to promise not to get their spouse pregnant. And so we really have to think of it differently because a lot of drugs cause mutagenesis, not just lenalidomide. So again, it's just because of the thalidomide thing. So anyways, that's number one. Number two is, yeah, we need better drugs. We need more effective drugs. We need to figure out how to use them better.
I think for me, the current major concern is the fragmentation of healthcare. If you get a prescription to a Walgreens, then every Walgreens in the United States knows the prescription. But CVS, they don't have a clue, and Walmart doesn't have a clue, and there's no sharing. And there are many patients who, for example, get their myeloma chemotherapy at one center, get orthopedic care at a second center, get bone building medicines at a third center, and see a cardiologist for an unrelated heart problem at a fourth center. And in some systems, there's a complete lack of communication between those four. And it's done by high-tech things like writing a letter to the other doctors. And given our system the inability for multiple systems to share, so that if a patient is seen at Memorial and Roswell Park and at Mayo, it would be nice if I could instantly see what Dr. Shah, Dr. McCarthy had to say. That's not always the case. And with the way electronics have been developed, this fragmentation where different healthcare providers don't consistently have access, I think is a great disadvantage for a patient because their expectation, if you ask them, is that everybody knows what everyone else is doing, and that regrettably is far from the truth.
Just to add to that, I just heard recently about a company that kind of addresses that. I haven't used it too much with patients yet, but it's called Myelo. And what they say is that a patient downloads the app on their phone and the doctor can send it to Milo. And then the patient can look at the prices at different pharmacies and get it sent to where they want to. So it doesn't have to, we don't send it directly to a pharmacy, but you send it to Milo, and then Milo, you pick the pharmacy you want. But that way, I think the cost comes, like patients can choose where they're getting a better price, but also what's convenient and not have to stick with one pharmacy the whole time. I haven't used it much yet, but I just heard of it. So I just thought I want to share it based on that.
So with the Q&A, or somebody, okay, with the Q&A portion, we're having some mic difficulties. So I'm going to just put this in front of your mouth when you ask the question. But think about questions. This is really the roundtable part of our roundtables. This is where you're an equal at the table with everybody else up here. So think about the questions that are burning. They don't have to be sequential. You know, just what is burning in your mind, and based upon what you've heard today, what do you have questions about? We also have some online questions, and I'll get to those as we go along. But get to like a, you know, like the auction thing. Just give me a little signal or something. No. We need it for the online audience.
All right.
I'll hold it.
Okay. This is a trivial question, but it should therefore have a quick answer. If one gets the Evusheld shot, which it seems as though all of us in this room should one also be getting the new booster?
The recommendation of 2 days ago from our infectious disease department is yes, keep 2 weeks in between. You should get both, because our treatments and your disease itself both limit your ability to build titers against vaccines, and the Evusheld overcomes that a little bit.
Yeah, just one caveat. You have to do vaccines first, then Evusheld. Because theoretically, Evusheld will block. I mean, your body will say, oh, I don't need to make antibodies. I got plenty against COVID-19, but it's only going to last for 6 months. So to Jens's point, Dr. Hillengass's point, you want to wait, like when if you got Evusheld, you want to wait till you're due to get it again, then get your vaccine, your booster, assuming you've been vaccinated before. And then you get the Evusheld afterwards 2 weeks later.
My 5th shot scheduled October 14th.
My 5th shot's tomorrow.
Thank you.
Yeah, us old people.
I've had all 5 shots and I had the Evusheld. When you say the Evusheld only lasts 6 months, does that mean we should get it again?
Yes,
Okay, schedule.
plain and simple. Again, the Emory study was very, very thought-provoking, because they showed that myeloma patients just don't generate as high neutralizing antibodies to virus as other patient populations. We think that might be bad, but we don't know. That's the other problem, is you measure an antibody titer. I don't know what that means, because we don't have long-term data to show how protective something is. We're pretty confident about Evusheld, and this is for the patient population where you may just not mount a good response because you're on active therapy, or myeloma itself can cause immune paresis, where you don't mount good antibody or T-cell responses, both B-cell and T-cell responses to vaccines or pathogens when you encounter them.
Oh, wow.
So for the general public, they're saying that this new vaccine is like an annual vaccine, like a flu shot. But for us, we kind of were recommended to get boosters after 4 months, before. If we go and get this booster, which for me would be my 6th, are we still supposed to get another COVID vaccine like 4 months after, or is this a 1 year, one-time shot? I mean, I've had the Evusheld too.
We don't know. It changes every month.
Steve hasn't made any recommendations. So right now, I think it's 5, and then we wait. I mean, there have major people who are making the decision, so no one can answer the question. Fauci said it is feasible, as he projects, that we'd be getting an annual COVID flu shot. That's not solid. And so right now, I think the correct answer is 5 shots. And then we wait, because we just don't know.
Now, of course, Rand Paul has other things to say about that when it comes to Fauci. Those are amazing interactions. But right now, I can tell you that group at Boston Children's just published a paper, now it's in mice, showing that they may have found a common antigen to COVID-19 that will allow for, just like you have with polio, one and done, or you get a booster 10 years later. We don't have that yet. So right now, it's like the influenza vaccine. It's like whack-a-mole, because every year, new variant comes up, wham, you got to have a vaccine. In fact, scientists spend all their time trying to anticipate which viral variants will be prevalent this coming year, and then they develop the vaccine for it. And some flu vaccines are better than others because they don't always anticipate which strains will arise. So I'm also getting my flu shot tomorrow. So, and my shingle shot. So we just don't know enough. So long answer to Dr. Gertz's point, we just have to wait and see. You got a follow-up?
Yeah, just to follow up to that. So, I mean, I had my 5th shot 3 months ago, but now, you know, the guidelines say if someone had their last booster more than 2 months back to go and get this new one for the Omicron variant. Are we allowed a 6th shot if we're immune compromised right now, according to the guidelines?
How'd you get 5 until now?
Because 3 is the first, though, 3 was the first regimen, and then 2 boosters.
It was actually 2 is the first, then 2 boosters.
But then they changed it.
Yeah, get the Omicron. To Dr. You should get it, okay? Because it's not at all the same. I mean, it's a totally different variant than the previous ones. Plus, a lot depends if you got Pfizer or Moderna. There's some subtleties, but yeah, if you've not had the Omicron, I would strongly recommend you get it. The chances are you won't. If you weren't vaccinated now, you would have a high likelihood of ending up in the hospital very ill. And in fact, in our allogeneic transplant population, our death rate was about 50%. It was horrific pre-vaccine. That's why it makes me ill just to think about people who, for whatever reason, say, I don't want to get the vaccine, I'm waiting. One of our football players, Cole Beasley, said, I'm going to do my research before I get the vaccine. Like, what are you talking about? But I do say that you should get and keep up with the vaccines, because it'll protect you so that even if you get COVID, you just won't get as sick. That's really the most important thing.
And just to verify that, Jenny had a CAR T transplant. She's on the CARTITUDE study and also had COVID. And because of her vaccines, it wasn't that big a deal. We have a question right here.
Actually, I've got 2 questions. First one, just with the CAR T, are patients only getting that if they're in a clinical trial, or is it something that you can try to get for a patient in your practice?
There are 2 products approved, but as we discussed earlier, the access to those is very limited right now because of the production issues. And they have certain criteria. And unfortunately, we have long wait lists where we basically try to keep patients alive and also in a good enough shape to get these CAR T cells. So at the moment, 2 are approved. There are studies going on. The studies sometimes have other inclusion criteria than most of the time have other inclusion criteria than the approved ones. So it's very specific to every patient, depending on if they're eligible for a trial, if they're eligible for the commercial product that would be paid by the insurance. Yeah, so there's all factors that we are considering.
Okay, and my other question is just clinical trials in general. I don't know much about them, but I know there's usually multiple arms. Are patients, when they go into a trial, are they like the placebo concept? Are there patients that in a particular arm would not get anything, or does each arm provide some therapy for all the patients in each arm?
So that's a good question. I think you're asking about generally clinical trials. And so scientifically, a trial cannot be done if one arm is so significantly inferior than the other, and it's going to cause problems to the patient. So it's usually a trial will have if it's a randomized study, each arm, like scientifically, we don't know that one arm is definitely better than the other, which is why the trial is happening. If we already know one arm is better, then the trial shouldn't happen theoretically. And so in the sense when if it is a placebo controlled trial, then we are thinking that placebo is the standard of care in that situation, and that's why it would be. But in myeloma, usually there aren't placebo controlled trials. They're usually with some other therapy in the other arm.
First of all, clinical trials are developed by global thought leaders. These are the smartest people in the world on myeloma as they sit down and meet on a regular basis to decide how to make things better for myeloma patients. The trials aren't developed to fail. The trials are designed to push the envelope forward and make things better for patients. That's one. Number two, the ethical regulations that underpin clinical trials and the federal oversight is staggering to ensure that everything is done ethically and appropriately. So for example, a trial of a patient with previously treated multiple myeloma, there'd never be a placebo because there are effective therapies for previously treated myeloma. It's usually comparisons of different groups. Clinical trials also pre-specify the monitoring that a patient has to have. And I've seen what these clinical trials do, and the monitoring is better than what occurs in general practice. And so I think patients are actually being scrutinized in a much, much better fashion. So this whole concept of placebo and I'm a guinea pig is a complete misunderstanding of trials, and two, intrinsic distrust of the medical system, frankly, that your doctor's not trying to do the very best possible for you. So when the COVID vaccine was introduced and no people were ever treated with it, it was completely ethical. You didn't know if the vaccine was safe, and you didn't know if it would protect against COVID. So there was a placebo arm to determine if, but as soon as it was clear that COVID vaccine helped, all the people on placebo were unblinded, were told they received placebo, and offer the treatment. So placebo only occurs when there is no proof that anything other than placebo is any better. Dr. McCarthy did the very, very first trial after transplant of using lenalidomide, Revlimid, or placebo to determine whether treatment after maintenance treatment after transplant was helpful. He showed it worked. Countless lives have been saved since that time, and it's become the standard of care. But the opportunity, I'll bet Jenny's been in a half dozen clinical trials. I'd be shocked because she searches out the clinical trials and gets herself registered. And at least at our center, we have more CAR T capacity in clinical trial settings than we do with commercial CAR T. So one of the strategies to access some of the new technologies or the newest drugs is entering a clinical trial.
Yeah, I second everything Dr. Gertz said, because at the time we developed that trial post-transplant, that was 2001. There was no standard. I mean, there was thalidomide, but nobody could tolerate it for maintenance. And that trial opened up in 2004, and we didn't read it out for a few years later. It took a long time. But yeah, it was very ethical to have patients on placebo because we weren't even sure that lenalidomide was going to be better long-term. And as you saw the cautionary tale about venetoclax, that's why we do trials, because venetoclax looked very promising in smaller studies, but actually inferior overall survival. That's why we do placebo-controlled trials. And then lastly, my granddaughter, who was 18 months old, had the opportunity to participate in a trial at Strong. So she got randomized to either a COVID-19 vaccine, she was on Pfizer, or placebo. So she got unblinded 6 months later when she was like 2 years and change. She had gotten placebo, then she got vaccinated. I had no problem with her being on that trial, even though she got a placebo, because it was important. To Dr. Gertz's point, we had to prove it works before you just do it.
Could you mention data safety monitoring?
Oh, yeah, yeah. There are such things as DSMBs, data safety monitoring boards. I participated in them as we all have. And what that is a group of investigators who are not part of the clinical trial, who have access to the data and see the data. It's usually done through a clinical research organization independent of the company, where we will go over data and then say, yeah, this is good, or oh, we have a problem here. And so that's really important, is that there is, to Dr. Gertz's point, there's tons of regulations to make sure it stays safe for our patients.
I just wanted to ask if you could address patients with high-risk disease as it relates to these new CAR T and the bispecific treatments, and whether there has been any new research on that.
Not specifically for high risk. We obviously in high risk patients where we know, okay, there's a higher risk that the disease comes back earlier, that the treatments are not as effective. There's a medical need, as we call it, or an urgent medical need. To my knowledge, I mean, there are obviously, we do subgroup analysis, but the numbers of patients who have been treated with these new treatments are not very high, and you only have a certain percentage of those patients being high risk. So it's not clear if a patient with high risk responds better to CAR T, worse to CAR T. That's not clear. Unfortunately, what we have learned in history is that standard risk patients respond better to any kind of treatment than high risk patients. It's very rare that a certain drug works better in a high risk patient that it would work in a standard risk patient. I'm not aware of any drugs. I'm not sure if you have any.
Risk keeps changing. We used to have ISS. We had ISS 2. Now we've got the revised ISS. And revised ISS 1 and now revised ISS 2. Sorry, misspoke. And chromosome 13 abnormalities used to be high risk when Bart Barloghi first classified them. He really pushed the field. They're no longer. Once bortezomib came out, it no longer was high risk. So a lot depends on time and understanding. There's a paper I'm reviewing now. I'm not going to say what, but it's a large study, and they did a lot of molecular analyses, and they think they may be able to figure out which patients benefit the most from lenalidomide maintenance. Now, it's an older study, but still, this is important. So the field keeps changing, and as we understand the biology of myeloma better, we're going to have a better idea of what is risk and what is truly high risk, and we may tailor things so that Dr. Hillengass's point, 11-14s may all need to get venetoclax early in their treatment because that may be the best for them long term. We don't know.
Hi, I just had a question about the CAR T. There has been a lot of discussion over it, and what piqued my interest was a couple of doctors. McCarthy, Gertz said it was, if I heard it right, overrated. And I was just curious if maybe we could talk a little bit about more of that and sort of organize those thoughts and maybe hear from Dr. Hillengass and Dr. Shaw.
You said overrated?
Yeah. No, it's not the panacea everybody thought it would be. And it's the same thing with CAR T for lymphoma. You only have about 30 to 40% of patients who have long-term durable remissions and probable cures. That means the majority of patients don't benefit from that CAR. I mean, they benefit short-term, it may control their disease, but it comes back. And so that's why this isn't like the Kaplan-Meier curve remains at 80 to 90%. This isn't childhood acute lymphoblastic leukemia, where the long-term outcome is 80 to 90% cure. We're not there yet. That's why I think people were all excited that this is going to change everything. It has for certain patients, but not for everybody. And I think it's even more difficult for myeloma patients, because myeloma patients, for ide-cel, the median progression-free survival is about 10 to 12 months. which is okay. I mean, these are people with bad disease, but still, it's not the same. It doesn't even seem to be as close to lymphoma. CARTITUDE, the Janssen Paradic cilta-cel may be a little better, but there's no comparison yet. And to be frank, in our own experience, we've had patients with both ide-cel and cilta-cel who relapse fairly early, and we have a few patients who are doing quite well, and we just don't understand yet who they are.
There's not much to add. It's so individual, and we are still in the process of learning, as we do with everything. In the patients in this room who got a high-dose chemotherapy benefit from the experience that they made 20, 30, 40 years ago. And the patients in 30, 40 years will benefit from the experience that the patients now go through in clinical trials, and that's why we do clinical trials. It's not, we don't know enough to say this patient will benefit and has no side effects. It's just not possible right now.
Just maybe one last thing in relation to your question is that in the CAR T trials, patients who got the CAR T cells had to have been able to get chemotherapy, get the collection, have their disease under control, let the cells be made, which take about 4 to 6 weeks. Then they have to come back in for the infusion and all of these things. So they inherently had a disease biology that could wait for that time. So there is some, you see that in the CAR T studies, they've done better, but we're also selecting for patients who are able to do this when we do CAR T cells. In the real world, we are able to push that envelope a little more and say little sicker patients, little faster disease, higher disease burden, we're going to put them on the CAR T. So we probably won't see the outcomes as good as what we're seeing in the trials, though that is yet to be determined. But a lot of it in the CAR T world is this self-selection, whereas that selection is not there in other trials often, because you can put them on the trial immediately when they are ready to need the treatment. So that's something to think about with CAR T, which is not with other therapies.
I just want to distill, I think, what all the other speakers have said. CAR T will shrink the myeloma when nothing else is working. And so one can't poo-poo that. But again, distilling what Dr. McCarthy said, the myeloma with our current products comes back in half of the patients within a year. And that's, I think, what most people don't actually recognize.
Now, there are other products. So, for example, there are allogeneic CAR T cells in clinical trial right now. And that's where they're really interesting. I do recommend reading any biography of Jennifer Doudna or Charpentier, the two women who describe CRISPR. Because CRISPR has allowed us all kinds of interesting ways to potentially genetically modify cells, including CAR T cells. So you can take any healthy person, you can knock out certain things that allow you to use the cells so it won't be rejected by the patient and won't cause something called graft versus host disease, where the CAR T would attack the patient. That would be bad as well. And then you put in a CAR, a chimeric antigen receptor against, say, BCMA, and that goes after the tumor. That's not ready for prime time. It's still in clinical trial. But that way you may get around to Dr. Shaw's point. You may be able to pull it out of the freezer and just use it for the patient. But the problem is it's still 101 because the majority of patients still progress. So we need to have a better understanding. Maybe we need dual CARs, GPRC5D and BCMA. Maybe we need to collect earlier. Maybe we need to do an allo or a CAR T and infuse it every month. These are all questions we just don't know yet.
With all of these medications and different cocktails, there is a potential for renal toxicity. What can we do as patients to offset that, manage that? What would be your recommendations?
Since it's very individual, it's also very difficult to answer this question. For a neuropathy, there are certain things we can do. For diarrhea, we can do certain things. for infection. A high risk is obviously the immunocompromised state of a patient on active treatment, because myeloma is a disease of the immune system, so there's collateral damage. So what we several times mentioned is vaccination is very important, being careful, using hygienic standards that are higher than for the general population. It depends very much on every single side effect, and it's very difficult to give a general answer to that. And you have to talk with your doctor, sitting in front of them saying, this is my side effect. And then your doctor can say, okay, this is what most likely will help you. And it's not even 100%, unfortunately, because one patient responds to a certain treatment and the neuropathy goes away. Another patient has no response whatsoever. We are very individual. Every single patient has a different set of response, side effects, duration of response. You can have a great response now, it can last very long, you can have a great response that you lose. So every single patient is different. That's what I meant with multiple myeloma. So every patient, and some patients have no side effects whatsoever, and that's great for them, but it doesn't help the patient who's sitting next to them in the waiting room who has a lot of side effects, right? And we try to learn more about it, and that's our job. But your job is basically to tell us what's going on. And I think it's also good to have things like HealthTree, where you can share your experience, because I have learned a lot, especially when it comes to side effects. I have learned almost most of what I do for my patients to improve their side effects from other patients who told me I have done that and it helped me, right? And that's a little bit try and error, but that's how we deal with that.
Most common cause of kidney damage in a myeloma patient is not the treatment of the myeloma, it's the myeloma. At presentation, 1 person in 6 with multiple myeloma will have impaired kidney function. By 6 months, 2/3 of those patients will have complete recovery of their kidney. And so you really have about 1 person in 20, 6 months into it, has persistent abnormalities in kidney function. Actually, most of the treatments, with the exception of stem cell transplant, have very, very little impact and don't damage the kidney, although adjustment of dosing is required when the kidney is impaired, but they're not really very harmful to the kidney. Transplant, that can cause changes in the kidney, and that's why people focus just on doing transplant, so they have experience to understand what are the things that need to be done if a patient who has impaired kidney function decides to proceed with stem cell transplant. But there's a lot out there in terms of support and expert teams to manage that. But if you're talking about the conventional treatments for multiple myeloma, they generally don't damage your kidney. If the kidney's starting to go bad, you worry about the myeloma being more active.
We have some online questions as well. And remember, this roundtables are like one-room classrooms. So we have novices and experts in our classroom, and the novice questions are just as important. And I'm sure you'll answer this one quickly, but it's asked a lot. Is there a way to pre-screen for myeloma?
That's actually not a novice question, because there's a lot of research going into that. There's actually a study going on in the country of Iceland where they do screen for myeloma. The good thing is that this will take a long time to read out, because myeloma still remains a rare disease. MGUS is fairly common. The older we get, the higher is the chance that something goes wrong in our plasma cells and we start to produce a certain protein, which we call monoclonal protein. So this is actually a good question, but we still don't, at the moment, we don't screen because myeloma is fairly rare.
Okay. Second, another question about maintenance. And I'll condense this. So with respect to things like Velcade or Revlimid used in maintenance therapies 2, 5, 10 years, when is it the best time to stop maintenance therapy? And is MRD, is minimal residual disease measurement, or are other algorithms a part of that?
It'll take about an hour to answer.
Exactly.
Well, first of all, most of the studies that were done with bortezomib were for 2 years, sometimes fixed number of doses, almost always. There were no studies that were done continuous bortezomib because it's too toxic. So the large trials, again, were about 2 years, and there was efficacy, but the problem is the HOVON 65 GMG4 trial. I said GMG4 because the Germans get always pissed off that we just say HOVON 65, which is Dutch. So that was comparing bortezomib to thalidomide. Well, that's stacking the deck right away because thalidomide's inferior. So that showed a benefit. And so then for a while, everyone's saying, oh, if you have deletion 17, you have to get bortezomib, not lenalidomide. But that's actually not true either. We have some other data that'll be coming out on it. So what we don't know is when to stop because lenalidomide does have toxicity. Some people have low blood counts. The British and the Italians did 3 weeks on, 1 week off. In the US, it's every day. So there's a whole bunch of people who will often tolerate it better if they get a different schedule. So I think the biggest problem is we don't know. Okay. And there are studies that are going to answer this question. For example, there is a SWOG trial. It's Dara-Len versus Len. And at 2 years in, you're going to get a bone marrow biopsy. And if you're MRD negative, you'll be randomized to stopping versus continuing. That's going to read out probably about 10 years from now. And I'll be really old. And then everyone would go, we don't do Lendara anymore because now we're doing something else. And that's the problem where we have to have surrogate endpoints for long-term outcome. And until we have those surrogate endpoints, we're going to struggle with what the right answer is because the trials take too long to read out. But there are studies ongoing to answer the questions that were brought up.
In terms of, Len discontinuation, there are 2 studies. I think there's one at MSK ongoing for lenalidomide or any maintenance cessation. And then there's one at the University of Chicago. These are small studies, but what they're looking at is if patients have achieved this MRD negativity or achieved a complete remission, and if they've been in this for a few years, is it okay to stop and monitor really closely? So I think we will see some of that data too coming out over the next few years. And that will also be helpful in making decisions around it, because now that we know that MRD negativity is important in terms of prognosis, and there are some patients on lenalidomide maintenance in MRD negativity for years, are those patients still needing the lenalidomide, or can we stop? And I think that's an important area that's being looked into.
Another question, is extramedullary disease considered high risk? And if you could explain briefly what extramedullary disease is.
Yeah, definitely. It is considered high risk. It's just harder to treat. That's part of it. And often as myeloma progresses, it tends to become more extramedullary or less secretory. So less of the myeloma protein in the blood and more in other sites. And so that makes it harder to treat. And one of the problems with that is that clinical trials often are looking at the secretory myeloma or looking at having an M spike. So as we get more extramedullary or non-secretory disease or disease that's oligosecretory or secreting less, then it's harder sometimes to get patients on trials, which hopefully will change as we design new studies.
You all know that myeloma is a cancer of the bone marrow. And from prior discussions, it's easiest to conceive as myeloma as weeds growing in your bone marrow garden. These weeds that grow in the garden need that garden soil to grow. In the laboratory, to try and grow myeloma, it's really, really hard because in the laboratory, you have dishes and plates, but you don't have that garden soil, that soil in the bone marrow. But myeloma sometimes can change. And when it changes in a more aggressive fashion, these weeds are capable of growing outside the soil. And so if myeloma appears in the lung or in the liver or in tissues that are not that bone marrow garden soil, that's referred to as extramedullary. The myeloma can survive outside the bone marrow. And historically, that really has been a big deal, that all of a sudden your myeloma grows outside your bone marrow, gee whiz. But like everything, what's good and what's bad depends on what treatment you have available. And as treatments begin to improve, things that historically were not good become easier and easier to treat. So whether extramedullary is good or bad is an always evolving concept based on how effective the treatments are. But in general, the principle of weeds growing outside of the soil and just popping up is generally considered different from the traditional weeds that only can grow within the garden and damage the overlying bone.
Editorial comment. I just posted a story today on our website, and it has a link to Dr. Gertz's, one of the versions that we have of Dr. Gertz's Weeds in the Garden talk. I highly encourage every one of you to watch it. We have 3 versions of it online. But you will, whether you're a novice or you think you know everything, you really should. Well, I say it's the equivalent in myeloma to like Beethoven's Ninth. It really is the best talk in myeloma. So I just happened to post it today, so I hope you all will access that. One more question about amyloidosis. The patient here is aware that there's more data and statistics on stem cell transplant and multiple myeloma. What's the latest on stem cell transplant with amyloidosis?
Amyloidosis is a complication that is seen in maybe 5% of myeloma patients, where the myeloma protein actually deposits into the tissues and causes organ damage. For the majority of people here, it's irrelevant. It's not happening. This is a very, very uncommon problem. 1/5, 1/8 as common as myeloma. But the standard of care currently, by consensus, by agreement among specialists, is that you should get chemotherapy as defined. It's chemotherapy. And currently, there's a 60% chance all the amyloid proteins will go away. And if that happens, nothing further needs to be done. But for patients who don't have it all go away, or at least go away 90%, and if transplant can be done safely, the safety profile is very different than myeloma transplant. And there are many patients who have amyloid where it would be unsafe to do a transplant, so it isn't done. But if you get chemo and you really haven't had a big, deep improvement in getting rid of the myeloma proteins, then stem cell transplant is done next in most centers for when it can be done safely, to try and drop those proteins down to the lowest possible level. So it's used, but the frequency with which it's used in the last 3 years, it really has plummeted significantly.
Completely agree.
And with that, whoops, got too many mics here. With that, we're going to close the morning session. We're going to say goodbye to our online audience. But before we do that, I'd like to thank our speakers. Dr. Gertz and Dr. McCarthy will be leaving us after lunch, and Dr. Hillengoss and Dr. Shaw, and then we'll also be joined by Dr. Moisich this afternoon. This morning's session was all about how to think about myeloma. This afternoon session is how to live with myeloma day to day. So I hope you enjoyed this morning. If your questions weren't answered, and if other questions come up, you know where to find us on the website. We'll be happy to find answers for you. But first of all, I'd like to thank all of our speakers for the morning session. It was really good. So I'll say goodbye to our online audience now. We'll be back in an hour with the second session. And for the rest of you, the lunch is going to be in the same room where the breakfast area was. I'm hoping it's all been set up because I haven't been out there to see. So I'm hoping everything is all set for us. We're going to take an hour for lunch. This is one of the things you will never, ever see at the meetings that I organize, and there's a reason for it, is I don't have patient testimonials generally up here. And one of the reasons I don't is because at the lunch period, you testify to each other and you inspire each other. That's really what you need to do. While you're here, it's all about learning about the disease. So thank you so much, and we'll see you back here in an hour, and I'll start rounding you up about 10 minutes before. Thank you.
Oh, yeah. Thank you. Be looking up. Yeah. I know you will. Yeah. For example, the non-sending blocks. And that's it's like picking your favorite side. And it's we go all every night with us together. We think it's I think so I was going to fight the cat and cell here. So it's simply so right now in the
Just from our where one side is clear, there is one case, but
yeah. And then initially it looks so missing, right? And then you find stuff like that.
and COVID hasn't helped. That we have the COVID issue, and then now all these. I feel now we're gonna need like infection. Do you not COVID?
No, I have to.
Oh.
No. You have the first one.
You're
Oh, I'm first. I just open both.
Yeah.
Wanna go through? You should be right.
You can see one time as long as it shows. I think everything else is
I don't know, because we have some people who believe it's taking off. And others will still Yeah.
Okay. I hope there's some food for you.
Sorry, I'm not, you may want to get somebody else. I know it's still a schedule for us, right? We have to do all the paperwork, but you have to do the same thing. I don't know. Yeah. Yeah. What do you mean? Well, you can find another job, okay? Just because they say we want to do it, right? Be frank and I'll tell it to you. Okay, that makes no sense. Sorry. No, I don't agree. So find somebody else to prescribe. Plenty of people who take so big. I don't know. You have to. Well, Dr. Lee prescribed yoga. Yeah. And this one is over. Yeah. Yeah, real. Oh, it's slightly different formulation. I mean, right now, Adderall, that's another derivative, but they're having in the gut. But no, I would say 15 is not a good, and provincial is a little bit better safety profile relative to it. I think your cardiologist is about to so good. Right? Yeah, let's see it. Hi. Yeah. Yeah, good. Thanks. Well, it's not fun. It's not good. It'll be well. Yeah. Yeah. Although I have some patients. I mean, they're in and out. In fact, there's some people who put all this time. And we don't need right. We don't have the footprint to do that. We don't have a it is, yeah, you have to, but there's a huge spectrum of people but this is not a huge spectrum. And we don't have to do that. Yeah. I'm glad you're doing well. Nice to meet you. Hi. Yeah, no worries. Yeah. Maybe. I mean, the long we have some people it came down to the not that sort of type. We knew about 2 months to years to sort of like that. That is not hopefully one time. No, thank you. My last cat. No, thank you. Well, no, I'm very lucky.
Before we get online, I'm really just about it. Anybody need more info? I can help with it. I had a goal that we could go out. But I don't know. This one, I think we got. I'll be right out. And one of the things, totally off the record, I have very good experience with our president. So this check is great. If you have $20, great. If you don't, enjoy the top one, take one, and see me get that. Okay, how are we doing? You know where we go? Yes. Okay. Make it. Okay. Oh, and I'll get it to you. I have it up. Oh, yeah, same. So we know what the other two? Did you do that? Can we go to the switch all together? Then you do that. I don't know what they do. We're talking about the math, maybe do better than I thought. So that's the next. Are we on? Oh, that's wonderful. Okay. I'll say real quick. I don't need to introduce Dr. Hillengoss. You all know how I feel about him. But as I said, this afternoon's session, I think is super important, and it relates to a lot of the work we're doing at the HealthTree Foundation. And living with myeloma is not just about surviving, it's about living and enjoying every day. And that's what this session is all about, and what you can do to help yourself and your fellow patients. And with that, we're going a little out of sequence. We're going to start with Dr. Hillengoss, And then I will do the interview after that with Nathan, and then we'll be back on track. So with that, I'd like to reintroduce Dr. Hillengoff.
Thank you so much. Yeah, I think that's actually a good topic right after lunch. I feel fairly heavy right now. So fitness is something that it came to me, this topic came to me. Well, I love doing workout myself, and my patients, when, as I said, when I started treating myeloma, it was many moons ago, and we did not talk about fitness, quality of life, or any of that, because we talked about how can I keep you alive a little bit longer. Now, we thankfully have many new drugs and can prolong the life of patients, and now quality of life is important, because I said our credo in our department is, as long as we cannot cure myeloma, we want to at least make sure that our patients have the best life they can live. Topics why exercise matters even a little bit, and that's something that Greg added, and I can tell you Greg actually lost a lot of weight, I think, right? You're really in great shape. I saw him in Utah hiking up a mountain like it's nothing. So that's pretty cool. He's really a role model. 40 pounds, see, that's awesome. I didn't ask you if I can tell anyone, but it was so impressive to see you run up this mountain in Park City. So that was nice. And then the 10,000 steps, the magic 10,000 steps. I will talk about that a little bit. And then stress, exercise, psychology, what's going on in our minds and combined with our body. I think those are very interesting topics. There are several challenges. If we talk about fitness in myeloma, there are challenges. Obviously, bone disease is one of them, right? When I started treating myeloma, and again, I say this sentence, patients with osteolytic lesions, I had no clue. Can they do workout? And some people here in the room had discussions with me about that. What can I do? How much can I lift? How high can I jump? And initially, we had no idea if that's okay, if there's a risk. As you know, myeloma causes a disbalance of the bone building and the bone eating cells. And the bone building and eating cells, they are completely healthy. They're just overactive, the bone eating ones, and the bone building ones are a little bit inhibited. And that leads to these osteolytic lesions and to the osteoporosis. In the worst case, it leads to fractures. This is kind of a scheme how it works, how these cells, oh, you know what, just brief interruption. I turn off these slides so that you can see the slides better. I'll be right back. A bit better to see, I think. Yeah, so this shows the bone eating cells, the bone building cells, and the myeloma cells, and it's kind of a vicious circle, right? The bone eating cells get nutrients out of the bone that then feeds the myeloma cells, and the myeloma cells stimulate the bone eating cells. So these are the things that we have to deal with in myeloma, and you know, we give bisphosphonates, we give denosumab to prevent that, but there are other things that we can do as well. So our goal in myeloma is, from here on the left side, you see an osteolytic lesion. Usually that should be all this light gray scale, right? That's a CT of a vertebral body. And then we give treatment. And in some patients, not all, again, we don't know enough, but we are getting there. In some patients, we see how the body starts to rebuild bone. Can you see that? There's this white rim around this hole in the bone. And that's something that we want to see in our patients. And obviously our treatment, bringing the myeloma under control, giving bisphosphonates, giving denosumab, that should help. But there are other things that we can do on top of that. Another thing is, and please don't read all that, this is only showing you, as we discussed earlier, the immune system of myeloma patients is compromised. Because the myeloma, if cancer develops in the body, the immune system was not able to stop that. Because every day in every one of our bodies, there are cells that go wrong, and then the immune system says, you're wrong, I have to kill you. If a cancer develops like myeloma, then there's kind of this gap in protection in the immune system. And these are scientific data where myeloma has an effect on the immune system, weakens the immune system. That's where the infections come from. Then obesity is also something that we know about. People who are obese have a higher risk to develop myeloma and have a higher risk to die from myeloma. So being in good shape is actually good for your survival chances as well, and not even to develop myeloma. But I'm sorry, these frames are a little bit off. But it has been shown that if patients are more obese, they have a high risk to develop myeloma. So starting with the bone, what can we do to improve bone other than with our treatments? And as you might know, if when astronauts come back from outer space and now they stay longer and longer in the International Space Station, they come back and they are osteoporotic. And those people, I mean, they are the fittest on the planet before they leave. And then they come back and have bones of a very frail old person, even though they're still in their 20s and 30s, right? So what they do is they do weightlifting. Well, there is no weight in space, so they have to be very creative. And so they have very interesting ways to deal with that. But it shows that they have learned that they have to strengthen their bones by working out, by using our bones, our body always, what we don't use kind of fades away. And in space, they don't have any weight on their bones. So the bone fades away because the body thinks, why should I invest in something that I don't need, right? And that's why they have to put pressure on their bones. And that's something that we can learn from them for multiple myeloma. Then there are different things that you can do. You can run, you can swim, you can do other things. Conventional is conventional in this. And so they did studies, not specifically in myeloma, but in other patients or in healthy controls where they measured the effect of exercise on bone density. And bone density, we want a highly dense and strong bone. And so they see that running seems to be the most effective way, or a combination of things seems to be the most effective way to bring the bone to redevelop. Swimming takes away gravity, so we have this effect almost like in space, where swimming is still a good thing, but it is not as effective as, for example, running. The problem with running, again, is that there's an impact, right? Every step that you run, there's an impact on your spine. And that's something that you have to discuss with your doctor, in most of your cases me, to be sure that this is not too much for your bones. And this is again a lytic lesion, and you can see if there's this, where this blue arrow is on the right-hand side, you can see that there's a piece of the vertebral body missing because the myeloma has eaten it up and has overactivated these bone-eating cells. And there you can see that there's a high fracture risk because there's no stability anymore, because bone is, as you know, very strong material. And if that is the case, we have to be very careful with things like running. Then coming to the immune system, there's really good, again, this is just kind of a picture to show you, there's a lot of things that are going right when you work out. Workout, physical activity has a beneficial effect on most immune cells. There, again, don't read all of that, but some of the cells, and we talked about the T cells, they can be, can kind of be woken up, can become more active, can be less exhausted. And that is something that we need for our immunomodulatory drugs to get good CAR T cells and all of that. So if we keep our immune system happy by working out, that's definitely beneficial. And these are the effects of exercise on the immune system. We don't know how much this works in myeloma, and the current study will investigate that. We have first pilot data that will be presented at our hematologists meeting, so I cannot talk about it yet, but I can tell you it's encouraging. So what we have done with the help of HealthTree is that we send out questionnaires on the HealthTree platform and asked how much, and maybe some of you have participated, how much exercise or physical activity have you done? How many hours per week have you done before you got your diagnosis? How many do you do now during you have the disease? And what would you actually like to do in the future? And we saw that there's a clear drop. Even patients with MGUS who should not have a problem, just knowing about the diagnosis, seem to have a negative effect on their physical activity. So they were less active, and almost every patient said, I want to do more than I'm doing right now. So there's definitely an urgent need to get more data. So we asked in HealthTree, we got feedback from 794 patients within a few months, which is awesome for the researcher in me, like the kid in the toy shop. That was really cool to get it very, very quickly, a lot of answers. So we had a lot of myeloma patients, some smoldering, some MGUS, median age of myeloma patients, and unfortunately, fortunately, I don't know, Caucasians seem to be more engaged in that. I would like to see more African Americans also to be engaged, because they have a little bit different immune system sometimes, so we would learn from that as well. you know, I guess most of you know the history in this country and also other countries, which is the reason why they oftentimes don't participate in clinical trials, but we do have some patients also responding on that end. Again, I cannot really show you the results, but in 3 months from now, I can show you. So stay tuned. And we have a new study now where we build on our pilot data. We have a prospective trial with 3 cohorts. One cohort is doing exercise remotely twice a week for 6 months. So we have learned it has to be quite some time. As you know, our body changes not that quickly. And the older we get, the slower our body changes. So that's something we will measure immune markers, we will measure bone markers, we will measure quality of life markers, psychological markers like depression, anxiety, and we want to see what the effect will be of this exercise. That's one cohort. The other cohort is intermittent fasting, because I said obesity is a factor. I have to admit, back in the day when patients asked me, I heard fasting is good for cancer patients, I said, doesn't matter, cancer cells just take what they want. There's actually data, literature showing, scientific literature showing that fasting, healthy cells can adjust to fasting state much better than cancer cells. So cancer cells actually starve when we fast. Should not be too extreme, because obviously our bodies can be weakened by that too, but there seem to be positive effects, and that's something that we look at in our cohort 2. And in cohort 3, only for newly diagnosed patients, we look if beta blockers that block the negative effects that stress can have on the immune system. We use beta blockers in combination with myeloma therapy. So these are the 3 cohorts. And the exercise is the largest because we have the preliminary data. The intermittent fasting is very early, so we only have a few patients that we can enroll, but please approach us if you're interested and participate. Briefly about the 10,000 steps, the large women's health study where they screened hundreds and hundreds of people, healthy controls, found out that there is more steps per day is better. And I get no money from Fitbit, unfortunately, but I use a Fitbit. You can use whatever brand you like. And it helps me to, because I'm a little bit competitive, so I like to be beyond the 10,000 steps. But what they actually found, the not magic number, but the number that's actually scientifically proven is 8,500 steps, which helped me because I sit my butt so much, because I have an office job other than when I'm in clinic, and getting up and walking 8,500 or more steps per day seems to be beneficial. You can see this benefit kind of flattens out. If you walk 20,000 steps, that's awesome, but it doesn't make a lot of a difference in this setting. So 8,500 seems to be where 8,500 and more seems to be good to be active per day. And as I said, these step counts seem to help. And here's just a show. They looked actually into other things than cancer, but it was beneficial to have this around 8,500 steps. And then also what we learned, and my wife should actually give this talk because she's a nurse and now a personal trainer like Lindy, who will train you later. And she looked into the literature for multiple myeloma as well. And again, this is not literature for myeloma. We are building this literature right now. But in other control groups, they found that if you sit for 8 hours straight, which unfortunately a lot of us do, and work out after, the workout basically does not do anything for your body. So this sitting 8 hours straight is so bad that after that, not even workout can save you, right? So what I do now, and that's another thing that my watch can do, it tells me every hour to get up and walk a few steps. And they did 4-second sprints, just not sitting 8 hours straight, but interrupting the 8 hours straight sitting with 4-second sprints, just getting up a little bit was helpful. And those patients had much more benefit from what they did later on with their workout. And these are really easy things to do. Just set a timer for once an hour, get up and walk a little bit. So that's another recommendation that we have. So why would we promote HealthTree here? Again, I get no money from them. I like working with them because Greg and all the team they're awesome people. But for us, if you enroll in HealthTree, and it's a little bit promotional, but I really believe in what I say here, it will advance the field. Nathan earlier asked me about a few things and said, hey, no one has ever looked into that because no one has access to so many patients. You heard from Jenny that it's 10,000 more patients. That's awesome. So if you enroll, you can decide which information you give, but every information we get is helpful for research. So you get also access, obviously, and you will hear more about that. And you are on close supervision, so you get feedback from people. You can connect with others and hear what they do. Yeah, and for clinical trials in general, as Dr. Gertz said earlier in our roundtable situation, he said you get much more attention. Obviously, the monitoring is much closer because the study demands it. So being in a clinical trial is good for you as well. And oftentimes it even reduces the costs because the sponsor of the trial pays for the drug, pays for the treatment. And for our research, obviously, to find the cure, we have to do clinical trials. We cannot do anecdotal stuff. We have to do clinical trials. We can create new hypotheses, especially HealthTree helps us with that because that's real world setting. So we know what's happening and then we can find patterns in this large number of patients. And we have easier access through HealthTree, and we can then approach them with interesting stuff that might not be interesting for a pharma company, but it is interesting for the myeloma community. So my recommendations from a fitness perspective, and I'm happy to talk about that in smaller setting later on. Talk with your doctor first if your bones are strong enough to do certain stuff. Set achievable goals, because if you say, okay, tomorrow I want to run a marathon, that might not happen. Maybe some of you, but not everyone, definitely not me. Get your steps in. It doesn't have to be 10,000, 8,500 and more is great. Aim for, that's the general recommendation, 150 to 300 active minutes per week, depending on how strongly active you are. That is something that you could do. And even a walk outside can be already part of that. Don't sit too long in one spot, but get up and walk and then sit down again. And combine aerobic and resistance training, because the resistance training helps with the bone strength, right? Don't lift too heavy because that's too harmful for the bones, but some might be helpful. And then my recommendation, register on HealthTree. Thank you so much. Yes.
Dr. Hillengass also does our technical work. So, you know, he's a man of many, many, many talents. One of the fun things I thought we'd do, we have really remarkable people working on the HealthTree Foundation staff. I always say I've always been cursed in life because the first job I had out of college was probably the best job I'll ever have in my life. And now I have competition for that. And one of the wonderful people that we work with or two of the wonderful people we work with happen to be married, Nathan and Lindley Sweeney. And Lindley and some of you may know Roz, who's our myeloma coach, director. I think that between the two of them, there is too much niceness going on between the two of them. They are just absolutely wonderful people. So as soon as Lindley can come up here, we're going to do these 3 times. We're just going to ask, she's going to lead you in a short little exercise that we want everybody to do before we move on to the next subject. And then Nathan and I will speak after this. So I will now turn it over to Lindley for her first exercise. Lindley?
All right, thanks, Greg. I passed out to many of you, not all of you, sorry, a exercise guide that reiterates a lot of what Dr. Hillengass just talked about. His wife, Michela, and I worked on this brochure that has some of that great information, including some exercises and description of their benefits and how to do them, of things you can do at home that you can add into those breaks that you do. If you did not get one, I apologize. I had limited that I could fit in my suitcase. So if you didn't get one, let me know and we will ship one to you. Okay. All right. So I'm going to ask you all, we're going to work from our chairs right now. I'm going to try and get some fun music going. All right. All right, so from your chair, I want you to bring your arms up overhead and bring it back down, bending your elbows, opening up your chest, trying not to hit your neighbor. All right, bring it up again and down, opening up your chest, breathing out. All right, sitting in your chair, I'm gonna have you lift up one knee. All right, can you all hear me?
Thanks. We've got two more of those for you. And if we could have Dr. Hillengoss please come up and just have a seat, and Nathan, and Nathan, here's the mic. And we're just going to have a little discussion about, I got a mic here. We're going to have a little discussion about something we do that we're really proud of and that we think is going to make a huge difference for myeloma patients throughout the world. On one, and hopefully it will lead to a cure. So let me first introduce Nathan. You know, Nathan, I don't know your title. What is your title?
It's whatever you want it to be. My official title is Manager of Clinical Research.
One of the things you should understand that's different about the HealthTree Foundation and how serious we are, thanks to Jenny, is to my knowledge, we're about one of the very, very few types of disease organizations who have PhDs on staff to run research projects. and supervise everything. And that's what Nathan is. Nathan's so I baffle him and he dazzles them with brilliance. And he's a really brilliant guy. And I wanted you to get to know what he does, because I think it's really important for you to know what we do behind the scenes and how hopefully this will make a great difference in your lives. So Nathan, I just like to start out with, what do you do? What do you see as the most important things you do? What are what's your mission?
So I think to explain that, I want to provide just a little bit of background, super brief. So I was diagnosed with cancer when I was 16 months. So just a tiny baby. I had acute lymphoblastic leukemia. Fortunately, I was around the time when VAMP, the chemotherapy, just started really being used, and I underwent radiation chemotherapy up until I was about 8 years old, and then I was pronounced cure, and I've been cure ever since. But I've always had this vendetta against cancer, and I've always wanted to be in the trenches with research and to fight against cancer in any regard. So it doesn't have to be specific to leukemia. I've done breast cancer, I've done colorectal cancer, I've done liver cancer, and now I'm here working on multiple myeloma. And so my goal is to find cure for multiple myeloma, for all cancers. And when I finished my graduate work at the University of Arizona, I did a postdoc at the Huntsman Cancer Institute. A postdoc, just in case you're not familiar, is just more work at cheap labor. So I did that for a year at the Huntsman. And when I got towards the end of that, I was looking for my next step. Where did I want to go? Did I want to stay in academia? And so I started putting feelers out there, and serendipitously, I came in contact with HealthTree, and I loved what they were doing. I loved that there was an enormous support group, and I knew that my mom, when I was younger, needed something like that, and it didn't exist. So when I heard that, I was excited. And then the idea of having this opportunity for patients to consolidate the information, and then asking permission that we could then query it and ask some profound questions that weren't being asked, was just, I felt like it was next level. This is where research needed to go. We were going at a pretty solid speed, but it was a little bit too slow. And so we needed to go, it needs to go a little faster. And I think HealthTree is that vehicle to take us a little bit faster, and our destination is a cure.
Explain how, if a patient gets a HealthTree, starts a HealthTree Cure Hub profile, how do you use that data for research? And also, how do you keep it anonymous? Because a lot of people are afraid that their medical information will get out. And we have I guess a fail-safe in that in HealthTree. If you could please explain that.
Yeah, so, I think for all clinicians and investigators, it's our sacred duty to just keep the privacy of patients. And so, first of all, we ask for permission that we can use your data. If you say no, we accept that. It's not incorporated in our research, not incorporated in our analyses or anything, and you still get the benefits of HealthTree. But if you do allow us to utilize your data in our analyses, you get assigned a number. And so we don't know any identifiable names, addresses, anything that can connect you to the data is removed. And so you get some number, and that's still, I don't want to downgrade that it's not important. It's very important to us. So it's once we have that data, it's kind of like if you think of Amazon or Netflix, where they'll give you a recommendation based on your history. And we want to be able to do that for healthcare. And so we can offer recommendations for patients recommendations that they can then, of course, take to their doctors. We're by no means replacing yins. We'll never be able to replace the yins or Dr. Hillengas, sorry. And okay. But we want to offer these recommendations based on this accumulation of health data. And so essentially, if you share your data, if you have a particular mutation, let's say it's some translocation or deletion, we can identify other patients that have a similar genetics as you. And maybe this person has gone ahead. Maybe they're 15, 20 years down the line. We can then look at what worked for them and propose that to you. And hopefully you can have that same journey or the same path and same success as they did. Now, since the field is always evolving, maybe they kind of had some side effects are a little bit too severe, or they wouldn't recommend that you go through. Well, fortunately, there's a new drug that could be used as an alternative, and you could still achieve the same outcome with fewer side effects. I don't know if I answered the whole question.
Oh, that's one of the, and I'll ask this question to Dr. Hillengas. One of the issues that we are trying to get across to people is the value. First of all, defining what real world evidence and data is, what the value of it is, and why this is a very, very different time to be using it. I mean, we didn't have real world data 30 years ago, really, at this level. So could you explain, what real world data is, why it is important, and how the HealthTree Cure Hub profile fits into that? Now, this is for Jens.
Oh, that's for me?
Yes.
Okay. So I have to know an answer to that. Okay, no, it's a completely different approach. When we do clinical trials, as we promoted earlier, we have a clearly selected group of patients. We have so-called inclusion criteria. We say this age range, this stage of disease, this number of prior treatments, and so on and so on and so on, right? So we have a very strict, limited number. But as I said several times today, myeloma is so different in different patients, very heterogeneous. And there's a huge number of patients who do not fit this specific question. Real world is taking everyone who agrees to it and then look at their data. There's no pre-selection. A little bit, of course, if we want to know what's going on in myeloma, we don't ask an MS patient, but we have much more access to the much wider field. And initially, oftentimes, real world data was frowned upon because it's kind of messy. You sometimes have missing data. There's the hemoglobin from 1984 is not there. And in a clinical trial, that would never happen because it has to be there. Otherwise, you get penalized as an investigator and so on. So these are two completely different approaches. And they're both very valuable because the real world data is what is actually happening, right? We can have our ivory tower approach with the clinical trials, and we need that. But then we have unfortunately, life is messy, right? And cancer is messy. So we can, and then we need big numbers because, as I said, it's very diverse. And as Nathan said, another patient, only if we look in tens of thousands of patients, we will find another patient who is similar to you. If we look in 10 patients, all 10 will be different. To have a chance to find 2 similar patients in 10 patients is almost impossible. And I have to be careful. I think Kirsten Moisich is already in the room. She's the expert on that topic, so I don't want to lean out too much, but we need these large numbers, and a clinical trial can only have such limited numbers because they're very specific, and then we try to extrapolate based on that data, and for that we need the real world data.
This is for both of you. What kind of questions can real world data answer that clinical trials can't or don't?
Many. It's a very challenging
I know it's
question.
but give us like 1 or 2 examples. Like, what's a type of study that might happen that would never happen under the old regime?
I think what we just discussed in the break is rare situations. Like, for example, what we discussed is non-secretory myeloma. I have a patient who is struggling with that because she cannot get into a clinical trial because clinical trials usually exclude patients with non-secretory myeloma, but they are out there. They are rare. It's rare that it happens, but it can happen. And with the real world, if you have 10,000 patients and don't care if they are 55 or 75, then you can look at 2 patients who fulfill this criterion. But to make a study for those 10 patients or 20 patients, that's almost impossible because no one will pay for it, because they say, I will not invest millions of dollars for 10 patients. I invest millions of dollars for 1,000 patients, right? And that's one example for rare situations. If you have a large data set, then there's a much higher chance to find these rare situations and get information out of that.
So some of the research that we've done with investigators that I think is like a really good fit or really good place for HealthTree to excel is we worked with Dr. Michael Thompson. He's at Aurora Health Center in Milwaukee. And he wanted to know if patients were getting revaccinated after treatment. It is a standard of care or a standard practice to revaccinate after treatment, specifically after a stem cell transplant. And so we proposed that question. And we don't want to know just a specific location. We want to know nationwide. We want to get a very large sample size or very broad sample size. And the vaccinations that we're interested in were flu, pneumonia, shingles, and then a broad spectrum antibiotic. And he found that, or actually the kind of the interesting thing is in just a matter of months, we had over 500 patients participate, which is wonderful. And so that's you guys. You guys are participating and helping answer these questions. And so what he found was that for flu and pneumonia, patients were getting revaccinated, but for shingles, patients weren't, or fewer patients were getting. So there was a high representation for flu and pneumonia, but a low representation in shingles and the broad spectrum antibiotic. And so that was interesting. That was informative. So we could take that finding and then address the field and say, we're doing a great job in this area. We could probably pick it up a little bit in this area. And that's one thing that that is a secret power of HealthTree is we can reduce the time it does to do a clinical trial or to ask these questions. So I'm going to use Dr. Hillengast as an example, but he conducted a study in Germany. I think over 3 years, you got roughly about 600 patients to participate in a study. And then he came over here and he did the same study through HealthTree. And in about 3 months, we got about 600 patients to participate. And so he was able to double his efforts or double the number of patients that participated in a fraction of the time. And so that's one of the superpowers is that we can accelerate this research because we can quickly reach out to the patients, offer this opportunity for them to participate. And the wonderful thing is that all of you are excited about research and you do participate. And our numbers are always just eye-popping for clinicians that we work with. And so it's wonderful. It's a huge thank you. And this allows us to really accelerate the research field.
The other, and this will be the last question on this, or comment, whatever. One of the, and tell me if I'm wrong on this, because I may well be completely wrong. One of the real weaknesses of medical research, as I see it, is what we call post-marketing. Like what happens after these drugs come on the market? They've been used a long time. Who's doing studies about what these things, so if you've taken Revlimid for 3, 4, 5 years, what does it actually do to you? And how does that compare to everybody else? Is that a good way of looking at real world data in the sense that those types of studies never occurred before, but real world data now gives us the opportunity to do those types of studies? Is that correct, or correct me in the way I should be corrected?
Well, I'll go first, and I'll just say, yes, I think you're correct in that in most research, they have to have some kind of ending date. And so they'll do a 2-year or 6 months, 2 years, 5 years, and they'll go to that point and then they'll stop the research, and there won't be there may not be a follow-up. Where HealthTree can come in is that we can do a longer follow-up. We can look at the patient data and look at 10 years, 15 years and more. And like, I think it was Dr. Gertz that said that, you know, since the field is moving so fast, looking at 10 years may not be helpful because we're already on to the next drug. But there's some of us that, or there's some patients that are still on the drug 10, 15 years down the line. And we can look at that and see what the effects are and outcomes and so forth.
I can just echo that. You are right. It's really the case. I mean, sometimes the FDA demands that they're so-called phase 4 trials. That is when the drug is on the market, that they ask the investigators, us, the academia field, to answer questions, follow up, but as you said, not that long. And it's very expensive to do such a study, and you get it not for free, but much faster and at a given time. You don't have to start collecting. You can just say, hey, if you're a patient who has Revlimid for 3 years, please answer this question. That would never happen. In other studies, you have to enroll the patient, and you have to follow up with them, and so on and so on. And in HealthTree, we have a patient who had it for 3 months, 3 years, not 30 years, but still, right? So those are definitely benefits.
And this is, this is one of the reasons we wanted to bring this up. We have entered an age where patients can help research as much or more than researchers can these days because of the data that you have. And I think HealthTree is a revolution that we don't quite yet understand how big it will be. But consider that there are research studies now that take place that take years and years and cost millions and millions and millions of dollars. And doctors have to go out and raise funds and all of that. But we offer them and say, hey, we'll give you this data for free. You figure it out. You ask the questions, and it won't cost you anything. And so even though we're doing small scale studies today, I can foresee the day in 4 or 5 years where we're going to be doing really big studies that are going to make a real impact on your lives, but we can't do it without you. And in your packets, and for those of you who are online, you can find this online, but in your packets, there's a form. If you want help to get a HealthTree Cure Hub, you can just fill that out, put it on the desk where you came in. Same thing if you'd like to be a myeloma coach and share one-on-one experience with coaches, or if you want to coach, even if you want somebody to help you, we can help you with that. And you also have an evaluation in the back of your packet. So be sure you fill all of those things out if you want to, and if you'd like help on the HealthTree Cure Hub, but especially the evaluations, and just leave them on the table where you checked in, and this will really help us. But please pay attention to what Nathan is doing, what Dr. Hillengoss is doing with us, and that will be a nice segue into our next segment, and if you wouldn't mind just moving the chairs off stage, and then I will ask Dr. Urvi Shah to come up. And one of the studies we've done, or is ongoing, is a nutrition study that Dr. Urvi Shah is doing at Memorial Sloan Kettering. And I saw her and Dr. Hillengoss give presentations at our Coaches Summit last month, and was absolutely, well, you guys were blown away with some of the things Dr. Hillengoss told you today. You know, what you learned today was doing a little bit can really help you a lot, which is not what we had been thinking for all these years. And now Dr. Shah is going to talk to you about your nutrition and what you can do to help yourself there, and what she has found in her studies. Thank you.
Hi, everyone. Good afternoon. I'm really excited to be here to talk to you all about nutrition, and it's a good thing you finished eating, you're full, and now you can listen. I think nutrition, one thing to remember is that when we are really hungry, we don't make good decisions because we really need to feel like we want to eat. So often nutrition is a lot of like thinking ahead, planning what your next meal will be, so that you can actually make informed choices and not like scramble to find the meal just when you are hungry at that time. So these are my disclosures. And I just wanted to start out with, in this era of COVID, everybody is like, should we do the vaccines and the boosters, and all of that's important, and I'm not taking away from that, but I think there's also something you can do in terms of empowering yourself with nutrition. And the reason I bring this up is because we don't really talk about it enough. And this is one study, but there is another one. And in the interest of time, I'm only going to show you one. But, in some of the HealthTree talks that I've given previously, the other one I have discussed too. But what you can see here is that this was only healthcare workers, but they did one in the general like another one's been done in the general population too. And what you can see is those who eat plant-based diets seem to have lower odds of moderate to severe COVID and less COVID as well. So there is something you can do on a daily basis, many times a day, in terms of changing the severity or likelihood of outcomes of how you develop COVID. And if you look, low carb and high protein diets actually had a higher risk of severe COVID or moderate to severe COVID. So actually diet can make a difference. And here's one example. So I think good nutrition is important for cancer at all stages. And, we think about it only in the survivorship setting, and I'm going to show you some data why we should change that and think about it differently. So in the survivorship setting, we think like, okay, to prevent the risk of other cancers, medical problems, and things like that. But also in the treatment setting, because in treatment setting, many times we can't give treatments because people have comorbidities or other medical conditions. So if you have cardiovascular disease, it might be harder to get the carfilzomib, or if there's diabetes, the steroid doses are affected and more side effects related to that. So there, during treatment, and then with immune therapies now, I think there's, having a robust immune system is more likely to, probably we'll see better responses that way. And there is probably a diet microbiome connection there too. So I think, and then the third space would be prevention, where we reduce the risk of myeloma coming back, but also prevent myeloma from developing in the first place for people with MGUS and smoldering or any other cancer in the future. So with that in mind, we developed like nutrition intervention research program at MSK. So we're doing studies around nutrition, microbiome, and the metabolism in myeloma. So I'm just going to talk a little bit about, BMI, diabetes, nutrition, and the microbiome. And, I think this is what food or supplements can I take is often the most likely asked question by our patients, and also the least likely to be answered because we don't have a lot of answers, or so we think. So obesity increases the risk. This is just a quick summary, but what you can see is obesity increases the risk of developing myeloma in the first place, and also increases the risk of progression from the precursor states to myeloma. And then post the diagnosis, extreme BMIs are also associated with worse outcomes. So there is some data around BMI and myeloma. So having a healthy body mass index can be important. And these are the multiple mechanisms by which it may affect it. However, patients that also lose significant amounts of weight unintentionally due to a myeloma diagnosis are 1.6 times more likely to do worse. So it's not just about, losing weight. That's not what I'm saying. We want to keep a healthy weight and trying to maintain it. So it depends on what stage or situation each patient is in. Diabetes is also associated with an increased risk of cancer. And we know that a lot of the US population is either diabetic or pre-diabetic, about 45% or so. When we look at diabetes and the risk, in general, blood cancers and myeloma, there seems to be a risk in some large population studies. What we don't know yet, and we're working on, is diabetes and cancer caused by the same risk factors? So like, diet or age or weight, genetics, the same thing? Or is it that diabetes, like what causes diabetes then leads to the high sugars, high insulin, and then leads to cancer? And that's the difference between what we think about as correlation or causation. And a lot of epidemiologic studies can only tell us about correlation, that we associate two things together. But how does it really happen? That doesn't come from epidemiologic studies. So we did a study with the HealthTree Foundation. This was, we published earlier this year, where we looked at patients' needs for nutrition, their perceptions and practices. And what you can see is that since their diagnosis, about 82% of patients have questions, 57% say their oncologist doesn't address it, and 94% say that if an oncologist gave recommendations, they made changes. So I know that patients are interested in getting this information if it's there. And patients are interested in such research too. What was interesting to me was that when we asked patients before and after their diagnosis if they've made any dietary changes, there was a significant improvement in diet quality in patients, where patients improved their consumption of plant-based foods and seafood and reduced their consumption of red meat and junk foods. And this was just, obviously it's self-reported, so there is that bias, but just to see that most patients did improve their diet after a diagnosis, and encouraging to show that people are making changes. And so this is the American Institute of Cancer Research, or the World Cancer Research Fund recommendations. And what you can see, this is general for all cancers, and only a third of myeloma or plasma cell disorder patients knew about it when we asked. So I think it's important to be aware of the guidelines that are already there, because this is based on a lot of research and evidence that's already available. So plant-rich diets and limiting sugary foods and fast food and red and processed meat. So then we're looking at dietary patterns and cancer. And this, I think, is interesting because there are 3 large studies. So these are population studies, like one in France, one in the US, and one in UK. And what they had huge populations of 40 to 60,000 patients. And what they looked at is, like, they got all these patients to fill out dietary surveys and said, what are the different dietary patterns in this population, and who gets more cancers or less cancers? And all 3 studies very consistently showed that those who are eating more plant-based foods have less cancer. And then we look at myeloma. So there are 2 studies in myeloma to date. One is the Nurses' Health Study, and health professionals study. And you can see that those who had healthier dietary patterns, which are shown in green, so those are healthier patterns, had lower risk of death after diagnosis. And those who had an unhealthy pattern had a higher risk of death. So there is some, diet can make a difference in the long-term survival too of patients. And then we look at the EPIC Oxford cohort. So it's the same one from UK that I showed you here. They actually looked at myeloma patients within their study. Granted, they didn't have a lot of myeloma patients, but they did show that there was a 77% relative risk reduction in patients on vegan or vegetarian diets compared to meat eaters in developing myeloma. So there is, again, a pattern, all these studies showing similar things. So very recently, we published a paper. This was in all cancers, but we looked at what are the mechanisms by which diet may be affecting cancer risk reduction. And so we looked at multiple different studies and kind of summarized it into this figure. And what you can see is that, the 2 common diets we looked at data for is the keto diet and the plant-based diet. And the keto diet we looked at because if you see, at the bottom of the slide I've written, there are currently in cancer 46 keto diet trials ongoing, 20 active, 10 completed, and 6 terminated, with 5 unknown status. This shows that a lot of these dietary studies and keto diets are not, being able to be completed because a keto diet is very hard to do. And there are only about 8 plant-based diet trials, despite the evidence I showed you on the previous slide, where actually plant-based diets are associated with reduced cancer risk. So we are seeing this opposite, where interventional trials are doing the opposite of what the epidemiologic studies are showing. And keto diets also may have some benefits through weight loss, inflammation, and insulin, and the ketone bodies that form that have anti-inflammatory capacity. It's just that doing a keto diet requires it to be not just low carb, but very low carb. And it's really hard to get to that state unless you're truly planning it for long term. And I think that's why keto diets are possibly, somebody could do it if they feel like that's the only way that, because they hate fruits and vegetables, or there's some reason, like there's a certain mutation or a reason that they need to do it. But I don't think a keto diet is a sustainable long-term diet that can be used for long-term, patients, and if you want to do it as a lifestyle. Whereas I think plant-based diets, meaning more plant foods in your diet, is something that inherently comes a little more naturally and can be done as a long-term thing, and seems to have more mechanisms by which it can target cancer risk reduction, as you can see in this figure. So this study is not in cancer or myeloma, but I think it's a very interesting study, and it's 9 patients only and 5 days on a diet, a fully plant-based diet versus a fully animal-based diet. And plant-based diets are obviously high in fiber, so you can see their fiber intake change significantly between the 2 diets. And what you can see here is they show the difference in short chain fatty acids. So butyrate was higher in those on the plant-based diet, and so was acetate, whereas the branched chain fatty acids were different. So this is in the stool, and we'll go into it a little bit more of why I think this is important. So short chain fatty acids have anti-cancer and anti-inflammatory properties, especially butyrate. So that's one of the mechanisms by which plant-based diets may be affecting cancer and inflammation. This is a study, again, not in myeloma or cancer, but it's a huge study of over 10,000 individuals. And what they showed is that if you eat more than 30 plant foods per week, I mean 30 types of plant foods. So I'm not talking about eating broccoli 30 times in the week, but I'm saying 30 different types of fruits and vegetables, or even beans, seeds, nuts, tofu, any plant-based food, even spices. So more than 30 times per week is associated with better diversity in the stool. And there is a lot of studies, and in the interest of time, I'm not going to show you, but there are previous HealthTree talks where I've discussed this, but higher diversity is associated with better survival, better outcomes post-transplant. There are a lot of studies showing that. So this is one way that you could help your gut microbiome and diversity, is by just eating a variety of plant foods and trying to get to that number of 30 and more per week. After I'd given a talk last month, Rosalind mentioned to me that some of the patients formed a group and they have a shared Excel that they count the number of plant foods they eat per week. And I was really happy to hear that, because it looks like they're making, positive changes through that. Another paper that we have that's, going to be coming out soon is looking at patients who are on maintenance therapy. So these are myeloma patients on lenalidomide maintenance. And what we did is we looked at diet, we looked at the microbiome, and we looked at their outcomes. So we were talking about MRD negativity. So we looked at sustained MRD negativity in these patients. And what we showed is that patients who had higher diversity in their stool had higher likelihood of sustained MRD negativity. Patients who had higher butyrate producers in their stool also had higher likelihood of sustained MRD negativity. And those who had higher butyrate levels in their stool also had higher sustained MRD negativity. So, and then we associated this with dietary surveys. And what we did is we looked at patients who were, so we showed that eating more healthier protein, so like plant protein, seafood protein, though I think the benefits coming mainly from the plant protein, because you also see it with the dietary flavonoids. But we see flavonoids are plant chemicals that are only found in plant foods. So higher dietary flavonoids are associated with higher butyrate levels, and also the same thing in so we were able to correlate diet with the gut microbiome, and then with potentially myeloma control long-term. While this is a small study, it's encouraging that we're still seeing a signal with such a small study. And I think that this goes to show that there is an opportunity to make some changes. Other things is in terms of myeloma development, there are lots of studies looking at obesity, diabetes, the inflammatory diet, altered microbiome as risk factors for development. But what if we could try to tilt the scale and see if we can make a difference in some of these markers? And so we decided to do a study. This was the Nutrivention pilot study, which opened to enrollment last year, and we've finished enrollment. It's a 20-patient study with patients with an elevated BMI over 25. So if they're overweight or obese and they have MGUS or smoldering myeloma, they were eligible. we would provide the patients, we ship them meals for 3 months, lunch and dinner, and we give them recipe ideas for breakfast and snacks. And we follow them for a year on the study with coaching and visits with a dietitian very closely. And in this time, we collected samples and analyzed some of this data. So this is some of the early data that I'm showing you, which we presented at the myeloma meeting this year. What you can see is that we had a nice distribution of different races. About 45% were minority, which is, I think, big for most studies. It's much, much lower. Also, we had a nice distribution of different BMI, MGUS, and smoldering, and a third were pre-diabetic or diabetic. This is the compliance data. And when we talk about compliance, I'm talking about plant-based diets, but not just junk food plant-based diets, but unprocessed plant foods. So we were able to get patients compliance in those 12 weeks up from being around 22% before intervention to 90% during the intervention. So patients did really make a change, and we see a feasibility here. And we also looked at things like BMI reduction. So we had a 7% weight loss or BMI reduction. We saw an improvement in markers of diabetes, so like A1C or glucose control, LDL cholesterol, insulin, IGF-1, total adiponectin leptin ratio improved, and adiponectin, leptin, insulin, and IGF-1, there are lots of studies. In the interest of time, I'm not showing you, but there are studies showing that these are associated with myeloma and myeloma progression. So we are able to show that we're able to change these biomarkers or markers that we know are associated with increased risk. We also looked at the microbiome, and you can see, as I told you, why I think butyrate's important. We show that we were able to improve this in the patient's stool and improve the number of bugs also that are making butyrate. We also show some improvement in inflammation markers as well. So with that pilot study data, we've worked on developing a few other studies, and this study is going to be opening through the HealthTree Foundation in a few months. And this is a study of smoldering myeloma patients anywhere in the United States. What we're going to do is look at diet versus different supplements and the effect on the gut microbiome. This is a study that's going to open at MSK and also in this month, actually. So if anybody's interested with MGUS or smoldering myeloma, please reach out. It's very similar to the pilot study where we'll be shipping meals and things, but it's going to be a randomized study looking at diet versus supplement versus placebo. And it's a large study. It's also eventually going to open at Emory in a few months' time. So depending on where patients live, they would only need to come to one of these sites about 5 or 6 times in the year, but we're happy to have patients from anywhere. This is the Nutrivention 4 study. This is a study in myeloma patients in remission on maintenance. So these are patients on either maintenance therapy, but what we're doing is that we have a study open looking at daratumumab versus lenalidomide as maintenance therapy. And we're going to give some of the patients on this maintenance the diet. So at 1 year time. So you have to be enrolled first on the maintenance study. And then if you are on that study, you have the opportunity to join the nutrition sub-study. So with that and a few other studies we are doing is looking at dietary patterns in patients and supplement use habits through the HealthTree Foundation. These surveys will hopefully be up in the next few months, and we'd really love your participation in them. So the more the numbers, it's the better the data we can have. So just to summarize a few things, healthful nutrition habits. I think carbohydrates have been given a bad name because of the refined carbohydrates, but whole grains are associated with reduced cancer risk and actually very important in a healthful diet. So it's important to have those because they also have increased fiber. So we need fiber in our diet, which come through plants, and no animal food has fiber. Protein, I would say prioritizing plant-based protein sources like beans, tofu, tempeh, and reducing red and processed meats, improving unsaturated fats, which come from nuts, seeds, fish oil, olive oil or avocados, and reducing the saturated fat. This is the Canadian food plate, which I really like because 90%, like actually only 12.5% of that plate has animal-based foods, and the rest 87.5 is about plant-based. And I think it's a very unbiased food plate. There are some things just like thinking about, calorie counting and calorie restriction, I don't think is a long-term sustainable way of weight loss or even managing a lifestyle, because that brings a negative association with food. And it's something that we're constantly like, oh, what can I not eat? But focusing on what you can eat and regular meal times, meal planning in advance can actually be really good ways to do this. And if you think about what 500 calories look like, this is quite important to understand calorie density. If you eat 500 calories of fruits and veggies, you're probably going to be full. But if you eat 500 calories of meat or cheese, you're probably not going to be full, because just fiber fills you up. So when I'm talking about plant-based diets, I'm not saying that somebody needs to be vegan. I'm not saying somebody needs to be vegetarian. I think it's important to understand that these diets can be very healthy if they are done right, but they can also be unhealthy if somebody's a junk food eater and eats only potato chips or desserts, and that could still be vegan potentially. So what I'm talking about is a whole food plant-based diet where we're talking about minimal animal products, and we're focusing on unprocessed foods, then that would be healthy. Just one thing to know is the fiber gap. So 67% of consumers, so like if we surveyed all of you or maybe even the whole country, 67% of people think that they're getting enough dietary fiber. In reality, only 5% are. So we really have a fiber deficiency in the United States, not a protein deficiency, which is what everybody's focused on. So things that I would say is it's important to be whole food, plant-based, whatever the diet you want to do, that's fine with me. But the 80-90% should be coming from plant-based foods. And again, when I say all of this, things to consider is you don't need to make the changes tomorrow. You don't need to make it immediately, but think about what stage of your disease you are in and how you can do it in a gradual way that's more sustainable and long-term, because we don't want to make these changes and realize like we can't do it and then go back to what we were doing because, oh, it's really hard to do this, I'm never going to do it. So small switches and substitutions are more likely to make changes than trying to make, just suddenly overnight saying, I'm going to cut everything out. So with that, I'll conclude that, obesity, we talked about diabetes, we talked about nutrition and how it's important in COVID, cancer, and myeloma, and the microbiome, where diversity is important, butyrate-producing bacteria are important. And in case you are interested in being a part of these trials, you could scan the QR code and send us your information, and we can reach out, or you can reach out to us at any time. With that, I'd like to thank everybody who's made this research possible, because I wouldn't have been able to do it alone. And these are some of the talks we have on the nutrition and wellness chapter that HealthTree has that I've given over the last few years. But in the interest of time, I've obviously not gone into all the studies and details, but if you're interested in knowing more and why we talk about this, these are some of the talks you could listen to.
Oh, no, you're fantastic. So we are going to take a 15-minute break. So I'm going to say goodbye to the online audience for 15 minutes, and we will be back with some more interesting information. We'll start out the next session with Lindley again. We'll do some fun stuff. So 15 minutes. See you in a little bit. 2:10. Copenhagen, Hanover, Wolfsburg. Oh yeah, Delta. The Scheußliche. Okay, Vito, how are we doing? Let me know, give me like a 5-second intro, please. Give me 5 seconds before we're on. And we'd like to welcome our online audience back for the concluding part of our second session. And we're going to begin this session again with Lindley, and she's going to walk you through some other exercises right now before we hear our final speaker.
All right, now that we've just sat down, I'm gonna have you all stand up again. All right, let's start with those marching. Get that blood. I think we're going to make this a part of all of our future ones. I was talking on the side because you guys seem to enjoy this. Thank you, Lindley. That was great. And we got one for the road for you too before she goes. One of the things that you as patients are often not aware of. And I always use the term. The reason I had Nathan up here and we highlight our programs is because I want people to see what's behind the curtain of what we do, because it's quite remarkable. And at cancer centers too, you have literally hundreds and thousands of people around the world, well, hundreds of thousands of people around the world doing cancer research who really don't see patients much, but they are the guys the guys and the gals working in the labs,
who are supporting the people in the clinics. And I thought it was really, really important for you to see that and to see how a PhD here at Roswell Park conducts her work and how to include you into her work. So with that, I'd like to introduce Kirsten Moisich. Thank you.
Thank you so much. I am so thrilled to be here. This is the first time in over 2 years that I'm presenting in front of real people, not a computer screen. And oh, I need slides. So I'm not going to talk so eloquently about science as Dr. Shah did just a few moments ago. I'm going to be a salesperson for my science. So I'm an epidemiologist, and the only good thing about COVID is probably that you all know what an epidemiologist does these days. That wasn't the case. But I really want to convince you to not only yourselves, but also to convince others to participate in epidemiological studies. So make sharing your data part of your routine and help out people like myself. And I'm going to start like very simply, like, so in science, we ask questions, we have ideas, we have suspicions that leads us to form a hypothesis, and which we then test in a scientific study. And so in cancer research, we don't go straight to the human models. We start out very simply in so-called in vitro studies, where we expose cells in a petri dish to something that we think might cause cancer or prevent cancer, and see whether or not the cells will grow or shrink. And once we have satisfactory results from these in vitro studies, we go into the mouse model. And my colleagues in the basic sciences kind of have the luxury to be able to inject a precise amount of a carcinogen or a chemopreventive agent into an animal that was bred exactly for that purpose. And so to mice, we can do a lot. We can make them fat or make them overeat. We can get them drunk, or we can make them exercise. But really, no matter how eloquent or elegant these animal studies or these in vitro studies are, mice are not humans. And at the end of the day, we need to translate these findings into the human model. And that's where we, people like me, come into place by the conduct of an epidemiological study. So again, it's the same concept. We have a suspicion. We think that a certain exposure causes a certain disease. Reaching out to the exercise intervention that we are very much interested in here at Roswell Parks. So we think that perhaps exercise is associated with lower risk of myeloma. We systematically collect data. We are not only interested in the experience of the cancer patients, but we want to compare that to the experience of healthy individuals. We do rigorous statistical testing. And since we are studying humans, we are very careful in the interpretation of our findings. So we literally ask an 80-year-old woman how many sexual partners she had. Or we ask a 50-year-old woman with breast cancer how much alcohol she drank when she was a teenager. Or we ask a 60-year-old man with colon cancer about his dietary patterns 10 years ago. And you might imagine that we don't always get it right. So the 80-year-old woman might not want to tell us how many sexual partners she had. The woman with breast cancer might not want to tell us how much alcohol she drank, or the man with colon cancer might just not remember how much fruits and vegetables he ate. So there's a certain amount of uncertainty, we call it misclassification in the data we collect. And that's why it's important that we not only conduct one study that examines an exposure disease relationship, we have to have lots and lots of studies. So my mentor in graduate school told me, like, it's not the first study that establishes an association that is important, it is the 50th study that confirms that association. So you're interested in myeloma. This is the epidemiology of multiple myeloma. These are the established risk factors. And Dr. Shah talked about obesity, which is pretty consistent. But when you look at the other established risk factors, age, gender, with men being slightly at greater risk, family history, having a first-degree relative with this dreadful disease. We know that African Americans are at significantly greater risk compared to Caucasians. And then, of course, the precursor to multiple myeloma, MGUS. But you can also see for most of, with the exception of obesity, none of these established risk factors are modifiable. There's nothing that you can do about it. Like, thank goodness we all get older. We cannot influence our gender and race, and even for MGUS, the risk factors are not well established. There's a suggestion that smoking might be associated with that. But aren't we interested in all of these? Like, you know, what kind of diet could I engage with? Like Dr. Shah very eloquently describe what you can do once you have the disease, but it would be of interest, I'm sure, of you and many of us to see if there are certain dietary patterns that prevent you from developing the disease. Is exercise associated with a reduced risk? Is smoking associated with an increased risk? There's a large body of literature that shows that regular aspirin use is associated with a reduction in many cancers, but it would be interesting to see if that would also apply for multiple myeloma. So the same applies for occupational exposures, environmental exposures. There is a large literature that shows that certain reproductive factors are associated with female cancers, such as breast cancer, ovarian cancer, and endometrial cancers. None of that is known for multiple myeloma. So that doesn't mean that it hasn't been studied, but in epidemiology, it's almost like for each study that shows an association, there might be another study that doesn't show an association. So the literature is quite inconsistent. So why do we know so little about the etiology of multiple myeloma? One reason is because we study people. So this is really kind of these today's random medical news. We might hear that coffee is associated with depression in twins. And you might see, like in the news or in the medical news, where you hear on the Today Show on one day that fiber is associated with a decreased risk of colon cancer. And then 6 months later, you hear on the Today Show that it's not associated with it. And that's because we study people. And it's also a matter that our science is very approachable. So if, you know, a study that shows that aspirin use is associated with a decreased risk of breast cancer, that's very understandable. And if you compare that with, let's say, a study from my colleagues in immunology that shows that upregulation of TGF-beta leads to a downregulation of IL-6. That's not very sexy, right? Like, and not interesting to the public. So that's why many of my colleagues from the basic sciences, they basically think that what we do is not important. But of course it's important because, as I said, at the end of the day, we need to validate and replicate findings from the basic science into the human model. So how do we study risk factors for cancer? We have two major tools. One is a so-called case control study or a retrospective study. And here we start out with enrolling people with cancer and people without cancer that are as comparable as possible to the people with cancer, to the cancer patients. And then we ask them to go back in time and recall risk factors. So again, we do ask the 80-year-old woman with ovarian cancer of how many sexual partners she had. We ask the woman with breast cancer about her alcohol consumption. And you can see that there is a lot of potential for misclassification that kind of muddies the water, and that then results in often the inconsistency of our studies. A much more elegant study design are the so-called cohort studies or prospective studies, which very rare and almost impossible to get funded in a single institutions. I think the cohort studies will only, the new cohort studies will only be conducted by government agencies. So here we start out with tens of thousands of healthy people. And then we assess their risk factors at the baseline when we enroll them, but then during the time that we follow them up, we update their exposure information and maybe add new exposure information. And then we compare the risk factor data that we have assessed during the follow-up among the people who develop cancer and those who remain free from the disease. So cancer, as you know, it doesn't just pop up. You know, most cancers when the cancer starts to the point when the cancer gets diagnosed, it can be decades. So the follow-up time for cancer is also decades. So while this is better, we don't have to worry about people not recalling their exposure correctly. It takes a long, long time to complete these studies, and they're also very, very expensive. So most studies in cancer epidemiology are the case control studies because they are very efficient. They can be conducted within 5 years, which is kind of the time period of an NIH grant. That's why all studies that get funded last 5 years. Another reason why we know so little is because it's hard to study rare cancers. And when you look at the incidence of, if you just look at the upper part of this graph, you see for the most common cancer in men is prostate cancer, and annually there are like almost 250,000 new cases. And it's pretty similar for the most common cancer in women, which is breast cancer. And thank goodness myeloma is a rare disease, but when you compare the incidence or the newly diagnosed patients per year, it's much, much lower in multiple myeloma. So that makes it a very challenging tumor to study. So myeloma doesn't make the top 10, and that's a good thing, but from an epidemiological perspective, it's a challenge to have enough people available to participate in our studies. So studying multiple myeloma risk factors is hard, and that's why it's probably a reason why a lot of epidemiologists stay away from it. It's hard to get these studies funded. It's hard to conduct them. So for case control studies, researchers need hundreds of patients and healthy people to participate in case control studies. So if we have a study that's, let's say, shows the association between aspirin use and a reduced risk of multiple myeloma, and it's based on 20 patients and 20 healthy individuals. That is not strong data to make recommendations. But if you see the same association in a study that includes 1,000 patients and 1,000 healthy individuals, that is the data are much, much stronger, and that's really then a motivation for further research. So for a case control study of multiple myeloma, it's impossible to do at a single institution. So we could not be doing it here at Russell Park alone. And another issue is that not everyone wants to be part of a study. I have to say that cancer patients are very generous with their time and generally agree to participate if their doctor lets them, and that's sometimes an issue. But the healthy individuals often do not want to participate. And the way we try to enroll healthy individuals is by calling them up. It's called random digit dialing. And so you can imagine like someone, you're like at home, it's cooking dinner, yelling at your kids to finish their homework, and then somebody calls you up, do you want to be part of a health study? And so most of our experience lately, most of the people just hang up on us. Nah, I'm not interested. And but then those who hear like, oh, do you want to be part of a health study? Might be the ones that I kind of call the latte sipping, carrot crunching, Prius driving, really health conscious individuals who have really, really good health habits themselves. And they think, oh, I would just love to be in a health study, you know, because that might reinforce their good health habits. But they might not be comparable to the cancer patients, right? Like who just, you know, so they're just kind of biased to a group that is unusually healthy. And then, as I mentioned, participants might not remember the risk factors correctly, or they might not want to tell us, you know, because they are embarrassed or they think it's none of our business. And that really introduces bias to the findings of our studies. So in cohort studies, and Dr. Shah was mentioning some results from a cohort study, I think it was the EPIC study, the researchers have to wait decades to have enough people who will develop multiple myeloma. So in Dr. Shah's example, there were 65 patients. That is not a lot, and that is not a lot to make sturdy statistical comparisons. So it's a real issue. That's where there's very, very few robust findings for the development of myeloma from cohort studies. So this is why I want you to be part of my studies, because it is, as I said, it is very, very challenging to conduct case control studies of multiple myelomas. I don't even think that 3 institutions would be sufficient. It's probably would be a huge effort, and that's why I'm so in love with HealthTree, and I'm so grateful for the efforts of Jenny and Nathan and many others at HealthTree, because it's an invaluable resource for epidemiologists and other scientists, because the numbers. You know, it's not only multiple myeloma, but also the precursor diseases. Dr. Hillengas and I, we are interested in an exercise intervention among smoldering myeloma patients to see if we can delay the progression to full onset myeloma. And it would be impossible to do it without a resource like HealthTree, where Nathan Sweeney is helping us with identifying the patients. And it's just an incredible resource. And it's probably hugely underutilized by the epidemiological community. So it's very important for grant applications. So if I can put in my grant that I have identified 700 patients with smoldering myeloma, and of those 90% have expressed interest in participating in an exercise intervention, that sells in a grant application, and that really will include the likelihood of my grant getting funded. And funded grants bring results, right? Like, so we can, that helps not only me and my career or the scientific community, but it really also helps your community of myeloma patients. So I'm pitching to you. I'm sure I'm preaching to the choir because you come here to Russell Park or participate online. So you are probably already participating in HealthTree, but I would urge you to encourage your friends, like whoever you can encounter to contribute to this extraordinary resource. And I'll be happy to answer any question if you. Oh, okay.
No, I'm gonna be up there. So let me. So think about your questions. I'll just put this in the middle. Let me. We are entering the final stretch. I know everybody's really pooped. And does anybody have any questions they'd like to ask? Oh, goodness. I'm sorry. I'm gotta work on my eyesight here. I know we have some online questions too. I'll just fold this because it's.
All right, thanks. Well, again, appreciate all the efforts from everybody. And this is just a question about diet. And I've been trying to eat more fiber and doing all the things like a good boy and exercise. But one of the things that I was curious about is, I just came out of transplant and I have to take all these other drugs, penicillin and all this. So like, am I wasting my time trying to get a good, microbiome by eating all these fiber if I'm killing it with antibiotics? I was just curious about that.
So great question, which I don't think I have a good answer to, where antibiotics do affect the microbiome. But I do think that there are two different things you're doing. So patients, like for instance, there are studies looking at patients post-transplant, and the diversity of the microbiome is associated with survival post-transplant. So I'm assuming all those patients on that study had got antibiotics too. But we're still seeing that there's a difference in survival based on diversity of the microbiome. So I think all that you're doing is probably helping. It may be that the antibiotics are not allowing you to let it, like it helping as much as it could without the antibiotics, but I think it's still helping. So I would keep it up and not change that. That would be my thoughts.
And it's never a waste of time to eat healthy for all kinds of reasons.
Well, I just made some pulled pork this weekend, so I beg to differ. I'm joshing you, of course. No, you are correct. This is an online question. Are there plans for any study on the effect of nutrition plant-based diet on time to relapse?
Yeah, that's a great question. So a little bit of that, there are some challenges around trying to doing that. What you're suggesting would be something where you're doing a study in the first remission until, like looking at the progression-free survival in somebody who is eating healthy versus not. So we are somewhat doing that with our Nutrivention 4 study, where we are giving patients on the daratumumab or lenalidomide arm 3 months of a diet and going to see the effects of it. What the challenge is that, the time to relapse or progression-free survival is not decades, but many years, approximately 5 on average. So trying to do a study where we're giving meals for 5 years is very difficult. But I think if patients make the changes and sustain them, then we can compare them to patients who don't. So that's what we'll be doing on the Nutrivention 4 study. So we may have some data in the next few years, but also the study that I showed you that, is in press just now, where we showed that patients who have more diversity and more butyrate producers are more likely to have sustained MRD negativity. That you could extrapolate that to say that's because we no longer sustained MRD negativity is associated with longer time to relapse or progression-free survival. So maybe that signal will be there in that study, but that study was small. And so that's why, and because the follow-up's not long enough, we don't have that data in that study yet.
Here's a newsworthy epidemiological question. Has any connection been shown between the aftermath of September 11th in Lower Manhattan and multiple myeloma?
I'm not aware.
So at MSK, we looked at these large looked at the World Trade Center cohorts, like the fire department patients, and showed that patients who have MGUS, firefighters who were exposed to 9/11 were more likely to have MGUS than those who were not exposed to 9/11. So there is a definite increased risk from the 9/11 part. But more recently, also in Blood Cancer Journal, we published where we looked at other populations. So firefighters is thought to have an increased risk because they are also exposed to fumes and smoke and dust and all of that. So is that what's causing it, or was it truly the 9/11 part of it? And what we did in that study is look at other populations like police and other first responders who are not firefighters because they don't have the exposure otherwise, and also showed that there is some increased risk. So 9/11 is associated, and there is also data on the mechanism from the Einstein or Montefiore group looking at it in leukemia as well, which was published in Nature. So there have been some studies around it, and yes, there is a link. So we see a lot of the patients at MSK through the 9/11 program.
Just as a quick editorial, because I was just thinking of this. Many of you have seen on the news when police go into crystal meth labs or something like that, and they put these spacesuits on when they go in. The origin of that policy began with a myeloma patient. There was a firefighter in Portland, Oregon in the 1990s who was diagnosed with myeloma. And when they did studies on them, they realized he was being exposed to so many different types of chemicals through his work. And they determined that for certain types of chemicals, we need to have protective gear for the people that go work in there. So remember that every time you see one of these people in a spacesuit going into one of these places, the point where that began, you can trace it back to a myeloma patient. Here's another nutrition question we're getting, which is good. I almost stopped eating red meat and chicken, but eat some fish. How can I get sufficient protein for anemia or to prevent anemia?
So anemia is multifactorial, and there is this myth around that, the only way to improve anemia is to eat red meat. And I don't think that is fully true because if you're iron deficient, yes, you need to have iron to improve your anemia, or if you're B12 deficient, you need B12. But sometimes the anemia is from other things, whether it's treatment related or something. So you're not going to just raise your blood counts from eating more red meat, which I think a lot of people do, thinking that that's what helps. But also plant-based sources of iron can also help. So it's not that if somebody's eating plant-based diets, they're not getting iron. There's enough iron in beans and things like that. And actually some of that iron there may be some benefits around that kind of iron as well. So to answer the question of what you could eat to get enough protein. So for protein, I think plant-based sources, I think it's quite important to incorporate beans, seeds, nuts into the diet, where I think in the US, beans are very rarely eaten and in a lot of the world now, because nobody thinks about that. But beans are very easy, like you open a can, or if you cook them, they're one of the cheapest foods. So in terms of like, people think plant-based diets are expensive, but beans are actually extremely cheap to cook and eat. So learning how to cook beans, or, you know, soak them overnight, sprout them and eat them, is one of the most, I think, very important things you can do if you're trying to transition to a plant-based diet, because it's not just about fruits and vegetables alone. And if you're trying to feel full and things, and that's what happens is some people do plant-based diets, just eat salads and fruits and then say like, I'm not full, and, you know, I'm losing weight and I don't want to do this anymore. But that's because they're probably not doing all the components of it or doing it right. Other foods also, like tofu, seitan, these are things that could also be incorporated.
We have a question here.
Yes, Jens, there was a study that I love that you brought to my attention recently, and I think you'll do a much more eloquent job talking about it. But it's the one about the subjects that were inactive for 3 weeks, and then they did a 30-year follow-up. I was hoping you could just give a brief synopsis of what that is and what we learned from that.
I'm not 100% sure. You mean the long-term sitting?
It was the one where there were 5 volunteers or participants. They got a baseline. They were requested to be inactive. Does that ring any bells?
I don't know the study. I'm so sorry.
That's okay. So they were asked to be inactive for 3 weeks, and then they took some cardiovascular measurements, and then they did a 30-year follow-up. And then basically they found that the 3 weeks of being inactive was more detrimental than 3 decades of aging. Does that sound familiar?
I don't know the exact study. As we discussed earlier today, there are obviously negative effects of being inactive. Sometimes that's not in our hands. I mean, our patients sometimes have to be inactive because of treatment or because of symptoms. I don't know the exact study. I would have to look. If you have a link, I'm happy to look at it and maybe comment in the next interview that we have together.
And
I'm surprised that would go through an institutional review board to ask people to be inactive. Yeah, probably not in the United States. Yeah. Oh, okay.
We've had a question. You've spoken a lot about intermittent fasting. Could you just explain sort of the mechanics of what intermittent fasting is and how to go about it?
Yeah, the studies that I have found on intermittent fasting are, again, very diverse. Where it's very known is in the Islamic world, where there's Ramadan, where they only eat between sun up and down. And that is something where there's a lot of information coming from, where they looked for health benefits. As I said earlier in the talk, there seems to be an effect on fasting makes healthy cells go in a certain metabolic state where they basically wait until there's nutrition available again, and cancer cells oftentimes have lost this ability. So there's kind of a starvation period, and the length of the fasting is not 100% clear. What is oftentimes used is the 14 and 10 or the 16 and 8. So depending on how long your day is, and did I calculate right? Yeah, I think that's 24. So it should be. I'm getting tired. So those are the ones. So there has to be a longer period of fasting than the time when we are eating, and it should also not be binge eating in the time when you eat. And I have tried that because I will offer it to my patients, so I have to experience myself. My wife did that too, and surprisingly enough, for me, it was much less challenging than a continuous fasting over a longer period of time, and then just starting eating normal again. So this intermittent fasting, I think it helps with being compliant to doing that, to adhering to it. As Uri said earlier, if we do something that's completely unrealistic, we will not do it because it's not realistic. But if we do something that we can do, and this intermittent fasting, in my personal experience, was really doable. And I love food. It works. So that's something, and there seems to be a beneficial effect. And we have to see how that pans out in myeloma patients. The study is going on. And if you want to come by, you can enroll. It's only 1 month that we do it. I was surprised that our nutrition colleague from nutrition said 1 month might be even worth it.
On just a personal note, I started the intermittent fasting about 2 years ago. And so basically what I do is I don't eat for 16 hours and then I eat within an 8-hour period. And I usually starts at, actually it's less than that. I usually eat between noon and 6. And it is really hard in the beginning for the first week or two. And now it's just normal. I mean, I don't get hungry and it's your body adjusts fairly quickly. And I have to say, it's been a big blessing for me. And with that, having shined the light on me at the end, I would like to thank our 3 speakers for the afternoon session. I think we learned a lot today. I want to thank you. You guys, you all can go. We're going to do one more last thing with Lindley before we go. So unless you want to do the exercises where they're up here, yeah, that would be fun. That would be fun. And I'll move these chairs, and then Lindley's going to do one last exercise, and then I'll have the last word.
All right, let's get ready and stand up. All right, let's sit back down. Sit down. All right, now stand back up. That's an exercise. All right, practicing sitting up and down in a chair is an excellent exercise for making it easier to get in and out of chairs, your car, whatever. So a lot of active movements that we do can help us in our normal daily lives. All right, we're going to
what I like about Lindley and Nathan is you can look at them and see that they practice what they preach. They are in such good shape, and they are really a great model for us at the HealthTree Foundation to show a little bit really helps a lot, which is something that I think is wonderful. I want to thank you all for coming. I've been involved in myeloma since 1998. I am not a patient. I got involved when my wife's stepmother was diagnosed with myeloma in 1996. So I like to say I don't know much about the science, but anything about myeloma that has nothing to do with science, I'm your guy. And it's just been remarkable in the 20 plus years that I've been involved with this to see the changes. Very, very few people like Dr. Hillengoss and the doctors here get to see things change as dramatically as they have during the course of their careers, and we expect them to change more. The thing that we always have to keep in mind, though, is the big picture of myeloma, which is getting fantastically better with every passing day, really doesn't matter to you as an individual, because you've got to deal with this disease day to day to day. And it's great that everybody else is doing much better, but you still have to deal with things. It is so important in myeloma for you to be educated about your disease. You know, if you get a headache, you don't go to a seminar to learn how headaches begin and should I take ibuprofen or aspirin or Advil or whatever, because, you know, that's a simple decision to make. This is not a simple decision. And in order to be the healthiest patient you can possibly be, you must be a participant in your care with your physicians and your nurses. You can't be a subject that lets them tell you what to do. You have to communicate with them. You have to negotiate with them. You have to figure out what's right for you, because what's right for you might not be right for the next person. So I hope you got a little flavor of that today. When we put together these programs, the hardest thing to do is to decide what to leave out. We left out a whole lot today. There's no way we can go over everything you need to know. My goal on these programs is that you learn a lot about a little bit. If you learn a lot about a little bit, you're going to be well off as a patient, much better than knowing a little about a lot. And so that's what we try to do with these. And I hope we did that today. I want to thank you for coming. I want to thank you on behalf of Jenny. I know she's really sad that she couldn't stay. And trust me, if she didn't stay, she's sick because she's a trooper. So again, thank you. Be safe on your way home. Enjoy the rest of the day. I think it's supposed to rain tomorrow. So all the exercise you learned about today, practice it tomorrow. And to the online audience, thank you for joining us, and we will see you soon again. Bye.
Thank you for joining us for today's HealthTree Roundtable for Multiple Myeloma. You can re-watch this meeting on our YouTube channel or by visiting healthtree.org. We'd like to thank our myeloma experts who have volunteered their time to help educate myeloma patients like you. We'd like to thank our sponsors once again for their continued support, and we invite you to attend future roundtable events.
The HealthTree Foundation for Multiple Myeloma provides a set of rich programs and tools for all myeloma patients at every stage of care. We put patients first in everything that we do, from the creation of new programs to the design of our tools. Our programs are meant to accomplish two goals: to support and educate patients and caregivers, and to advance a myeloma cure. We've created remarkable programs that we hope help you become a better self-advocate so you can obtain your very best outcomes. As a myeloma patient myself, these programs are what I wanted when I was diagnosed. I hope you can take advantage of these very valuable resources to successfully navigate your myeloma and to help us accelerate a myeloma cure. The HealthTree for Multiple Myeloma news site is a news feed of relevant information about myeloma in patient-friendly language. We are lucky that so much development is being done in myeloma, but that also means we need to stay up to date as things are changing quickly. Subscribe to the weekly newsletter so you can follow major advancements. Read patient stories. Use the search button to search over 10 years of information about myeloma. Find a myeloma specialist for a second opinion in our directory, or learn from additional resources on this valuable site.
The goal of our HealthTree for Multiple Myeloma community events program is to connect, educate, and support patients and caregivers on different aspects of myeloma. We have chapters or different groups based on topic or geographic area that meet regularly online. For example, we have geographic chapters in Southern California, Florida, and the Northeast, Southeast, and Mountain West regions of the United States. We have chapters that meet based on topics of interest like fitness and nutrition, immunotherapy, or even stages of diagnosis like a chapter for newly diagnosed or relapsed refractory patients. We have chapter groups dedicated to rare forms of myeloma or related diseases like amyloidosis, non-secretory myeloma, and plasma cell leukemia. We also have chapters for African Americans and patients that speak Spanish. Forming these groups creates a powerful bond, a larger network, and incredible research opportunities as we join together virtually and occasionally in person once a month. It's a privilege to get to know so many myeloma patients, caregivers, physicians, and supporters intimately and support them in their journey. I absolutely love my job.
HealthTree Myeloma Roundtables are live patient education events featuring top myeloma experts. They're designed to provide in-depth and accessible overviews of the landscape of particular myeloma issues for novice and veteran patients. Myeloma Roundtables teach about particular topics in myeloma, not just basics. They help put things into context for patients to learn and understand more about their disease and how to best live with it until cures are achieved. Whether you're newly diagnosed or relapsed refractory, want to learn about myeloma testing, genetics, side effects, or the latest research and cutting-edge therapies, Myeloma Roundtables are for you. Each has moderated discussions, and the best thing I think about our Myeloma Roundtables is that they always have extensive time set aside for the audience to ask questions of the full faculty. This allows for interaction among the speakers and often provides insights that can't be found in papers and studies. Our goal is to help you ask the best questions so that you can have more productive visits with your healthcare team. We host 6 roundtables each year throughout the nation, and we live stream, record, and post each event on our website. We call them roundtables because we believe everyone touched by myeloma, patients, caregivers, family members, researchers, physicians, nurses, and anyone else who comes into contact with myeloma patients deserves a front seat at the table when it comes to the best diagnosis, the best treatment, and a realistic path to a cure.
Whether you are a caregiver or a myeloma patient, the HealthTree Myeloma Coach program is designed to help you find one-on-one support to fit your needs. You might be looking for emotional support, more information about myeloma, or just someone to talk to. No matter what you're looking for, coaches are trained volunteers and offer their personal experiences and resources to help. You can find a coach in your location or with specific areas of experience that would be helpful to you. You can connect in a way that works best for both of you, by phone, email, video chat, or in person. You might even want to become a coach yourself to help someone else on their journey. Orientation and training are provided to the coaches using our extensive HealthTree University curriculum, so answers to common questions are never far away. It's the personal experience that sets HealthTree Myeloma coaches apart. Having myeloma can feel so lonely and isolating. When my husband was diagnosed with myeloma over 7 years ago, I wanted to talk to someone who was close to my age, balancing children at home, working, and caregiving, to learn how they did it and what had helped them. I wanted to talk to someone who could personally relate to what I was experiencing. The HealthTree Myeloma Coach program can provide these types of connections. Knowing there are others who can relate can make all of the difference. I love leading this program because it truly helps us meet people where they are, provide personalized resources and support, and shows patients they can live a happy and full life, even with a myeloma diagnosis.
Educated and empowered patients tend to have better outcomes. HealthTree University for Multiple Myeloma is designed as an online myeloma curriculum to meet all myeloma educational needs. Whether you are a newly diagnosed myeloma patient or a myeloma patient expert, HealthTree University includes dozens of online courses with hundreds of lessons. Each course consists of multiple video lessons focused around a single topic. These lessons are taught by the myeloma experts who are world-renowned and treat hundreds of myeloma patients each year. They are the leading researchers in the field, with their findings being published in well-respected medical journals. The lessons also include video animations and graphics to help make difficult concepts easier to understand. Quizzes follow each lesson to evaluate the understanding of the topic. HealthTree University is continuously updated as the myeloma landscape changes. We are constantly interviewing myeloma experts at our roundtable meetings and key myeloma academic meetings to keep the curriculum up to date. When I was diagnosed with multiple myeloma, I believed in Doctor Knows Best, and I blindly followed my doctor's orders. As I've become more experienced, and as a former teacher, I realized the importance of arming patients with the information in HealthTree University. I want you to make informed decisions and become your own best advocate.
HealthTree's Black Myeloma Health Program is aimed at increasing awareness, education, outreach, and advocacy in our Black communities. I understand the need personally as a myeloma caregiver to my husband. We offer help and support for those who are on this myeloma journey, a blood cancer that African Americans are 2 to 3 times more likely to develop. We also have a higher incidence of the precursor conditions, MGUS and smoldering myeloma. Our programs aim to promote awareness, education, outreach, and advocacy to this patient group. We are doing this by creating a blackmyelomahealth.org website, teaching the Black community about the signs and symptoms of myeloma, taking our programs and events into the Black communities where health and wellness education is perhaps not free-flowing, partnering with Black organizations to perform greater community outreach and awareness, growing our African American Facebook group and HealthTree community chapter, and creating education in HealthTree University for the Black community. Taken together, we strengthen our African American communities and we effectuate positive change.
The HealthTree podcast for multiple myeloma was our very first program that connects patients with myeloma researchers. In the podcast, I interview top myeloma researchers about key myeloma topics and about clinical trials. Finding and joining clinical trials is not an easy task for patients. We need to understand what trials are open, who can join, and which patients are the best fit. So to date, I've interviewed over 150 myeloma experts to connect you with their open clinical trials. The program's been viewed or listened to by over 2 million readers and listeners and continues to be one of our most popular programs.
Exercise has many benefits for patients, including reduction of fatigue, decreased fall risk, improved mood, and overall better quality of life. One of the most important benefits for patients is that being fit can maximize the number of treatment options available to you. Our HealthTree Moves program was created to build community and increase movement. Throughout the year, we have a range of fitness challenges in our mobile HealthTree Moves app that is designed for all fitness levels with the goal to stay accountable and continue or increase activity. We have found through these challenges that participants are motivated to hit the goals they set for themselves because they have a group of others to do it with. We are also expanding our HealthTree Moves program with a growing 5K and 1 mile series, so watch in your area for an upcoming event. Our goal is to bring awareness, build community, and get moving together.
HealthTree Cure Hub is a patient tool that helps you navigate your myeloma and at the same time accelerate a myeloma cure. We built HealthTree Cure Hub to provide you with easy to use everyday tools that will help you make better treatment decisions in partnership with your healthcare team. In Cure Hub, you can track all your myeloma labs in a single place so you don't have to create or maintain a separate spreadsheet. Cure Hub can help you look ahead and find a list of treatment options personalized just for you that you can consider based on your prior therapies, genetics, and other factors. HealthTree Cure Hub can also search through hundreds of open myeloma clinical trials and narrow the list down to only those you are eligible to join. You can find and connect with patients that have similar experiences or genetics using our twin machine technology. You can also use our side effect feature, where you can see hundreds of side effects and thousands of crowdsourced solutions provided by patients. Learn from your peers to see what worked and what didn't to manage the most common myeloma side effects. I encourage you to sign up for HealthTree Cure Hub and investigate all the tools provided to help you make the best decisions possible.
More than 11,000 patients have joined HealthTree Cure Hub to contribute to research aimed at accelerating the cure for multiple myeloma. Because so many patients like you have contributed your myeloma story, we can now use this real-world data to reach faster conclusions and answer key questions such as, what are the best induction therapies? How are patients making treatment decisions at relapse? What are the best treatments for specific genetic subtypes? What are the outcomes of novel treatments like CAR-T therapy?
Patients have pressing health questions. Therefore, we need a practical way to help inform these key treatment decisions today.
Our HealthTree for Multiple Myeloma research program partners with investigators to answer critical medical questions.
We facilitate the ability to run queries on anonymized data and enable investigators to ask you questions.
We would like to thank you for contributing and responding to these surveys and participating in the research.
Because when you do, you improve clinical care in the immediate future.
If you haven't already, please join us in HealthTree Cure Hub to contribute to answering the critical questions myeloma patients and researchers are asking today, and to help us drive faster to a myeloma cure.
As we identified the tools you needed to successfully navigate your myeloma, we realized that third-party software was not going to be ideal. It was too disjointed and it didn't work the way you wanted it to. We have now built over 10 integrated software tools to support myeloma patients and caregivers. Nothing like it exists for any other disease, anywhere. To make it easier for you, each tool is part of a software suite where you can create one account using single sign-on. We use the latest technology and security methodologies to ensure your information is safe. Our development team of over 12 members ensures that we will continue to support you as we add new products and services that we roll into the HealthTree platform. We listen and respond to your input, so please feel free to share your thoughts so we can make the platform even better over time.
HealthTree for Multiple Myeloma has a diverse team of patient navigators who are always happy to help by phone or online chat. For newly diagnosed patients, knowing where to start can feel overwhelming. Our team can help you understand your disease, your type of myeloma, and suggest questions you may want to ask to your care team. We can help connect with a myeloma expert and explain HealthTree resources that can help you navigate your individual situation. For relapsed and more experienced patients, we can guide you through resources you may have not known existed. For example, understanding and keeping track of your myeloma is an important way to better advocate for your own care. Our team provides support for HealthTree Cure Hub, a tool that helps you navigate your myeloma while also helping move myeloma research forward. Our medical experts can help you gather all of your medical information into a single place, even for multiple facilities. This helps you keep an eye on your myeloma markers and track your key labs so you can more easily watch for relapse and ask better questions in the clinic about your next potential therapy. Our team includes African American patient navigators and Spanish-speaking patient navigators to provide more personalized support for those who may need it. Let us help you prepare for upcoming clinic appointments or better understand your disease by calling 800-709-1113. As always, we're happy to help.
For more information about our programs, visit healthtree.org.